This conference will now be recorded.
Thanks, Maya. From Southwest Florida, we have Barbara Welsh as well, and over on the East Coast from the St. John's River Water Management District, we have Chuck Jacoby, along with multiple people from the Tampa Bay, Sarasota, and Charlotte Harbor. We appreciate all of your input. One of the things I'd like to get out of this meeting is potential options for continued collaborations and increased collaborations. We're all doing wonderful work. We demonstrate it time and again, but there's always different ways that we can work together, try to get similar products coming out that we can compare across the systems to help each other in the management and improvement of these different activities. Go ahead and stop sharing my webcam because y'all don't want to see my mug. I'm going to turn it over to Marcus Beck now, who's going to talk about some of the latest open science app that he's been working on.
Do we want to go through and give intros to everyone? Gary first? Or no? About 60 people on the line.
Yeah, I don't think so right now, if unless the group would like that. It's always cumbersome when we're on like this to me.
So be it. Let's press on then.
Okay, turning it over to Marcus.
All right, thanks, Gary. Well, I can introduce myself. So I'm Marcus Beck. I'm the program scientist for the Tampa Bay Estuary program. And I know we have a lot of speakers today, so I will try to keep my time short and make sure that everyone has plenty of time to go over their talks. What I wanted to do essentially was kind of twofold. I first wanted to show a dashboard that's fairly new and maybe helpful towards understanding trends, specifically in Tampa Bay, but could be extended to other locations. So keep that in mind if you're not from the Tampa Bay region. This is something that you could use with further development of the product. But I also wanted to talk about, in the bulk of my talk, kind of a deeper dive into exploring some of the recent changes that we saw with the recent update to the seagrass layer, specifically looking at comparisons between this year's layer and the layer from 2018 to see if maybe we could do a deeper dive. And before I talk about all that, I will sort of preface that this is totally exploratory and meant to stimulate discussion and not a definitive analysis. This is sort of me kind of playing in my sandbox and sort of hoping that some of these analyses are helpful in terms of furthering our understanding of what could be potentially driving these changes. So I will go to presenter mode. There we go. Okay, so obviously, yes, this is motivated by the recent change that we had this year with the update to the 2020 seagrass layer. It's no surprise to all of us that we got a big hit in the bay, dropping down to about 34,000 acres. So a huge reduction from what we saw with the last seagrass layer. And so again, our motivating idea here is to really explore these changes in a little more detail. And to kind of put these into context, I wanted to show you all this app that we put together. This is a new one. We have several Seagrass apps that we've put out at the Estuary program to help us really understand the data in a little more detail, looking at the Transect data, the aerial coverage data, but this one particularly is just summarizing acreage estimates, or if you want, hectare estimates for Tampa Bay by major areas. This is actually showing the regional boundaries that the district uses and summarizing changes that way. We also have management areas that we use internally within the Estuary program based on areas that are more relevant for sort of more focused spatial analyses. But essentially, you can select different year comparisons and look at what was the change by, in this example, 1988 to 2020. And so you could look at what the overall change was, percent changes and how they break down by these different polygons, and then looking at how they map spatially there. So, you can just sort of slide around and look at different year pairs that you want to understand what those changes might have been, because I know it can be tedious to sort of download those large polygon layers from the district and try to do this yourself. So that was kind of our motivation here by providing those estimates. So have a look at that. And I said also that, this is something that we could extend to other areas, not just Tampa Bay. We have data for much larger parts of the state. So if you have an interest in this type of product, let me know, and maybe we could try to work something out and sort of apply this tool to other areas. So that's the dashboard. Again, be happy to talk more about that later, but I just wanted to put that out there as something that we have produced that might be helpful to understanding the different products. But this talk today is really more about understanding kind of, again, what happened between 2018 and 2020 with the seagrass layer. Again, going back to our management paradigm we think about for the bay. Obviously, we're all familiar with this, but essentially, we've seen the recovery of seagrasses over time by getting a handle on controlling point sources of pollution, so reducing nitrogen loads, what that meant for improvements in water clarity through reductions in chlorophyll, and of course, seeing the seagrasses rebound over time. So I wanted to sort of look at this paradigm and think about specifically the 2018 to 2020 change and really evaluate one particular hypothesis, the idea being that if water clarity is the driving factor, can we sort of look at that as something that could be influencing the changes we saw from 2018 to 2020? So sort of testing that hypothesis within the broader management paradigm that we're familiar with. So I did this, as you'll see in a minute, through some spatial analyses and trying to relate this to some of the water quality data we get from our long-term monitoring programs. But again, I just wanted to look at just these 2 years, the 2020 and 2018 data, to look at just this one particular hypothesis to see if this was an issue. Is water clarity a concern here driving these changes? So in the guise of like conventional scientific analysis, we have our null hypothesis that, if light issues or light conditions aren't a factor, where we see seagrass gains and losses, if light conditions aren't a factor, then maybe we might expect these changes not to vary as a function of depth. So essentially, if light conditions are an issue, we would see seagrass losses at sort of deeper areas at the maximum depth of growth there, the deep edge of seagrasses, and sort of see them receding towards more shallow depths. And so that would be my alternative hypothesis here, that if light conditions are a factor, we would see seagrasses growing in more shallow depths in 2020 as compared to 2018. And again, this is totally exploratory. This is me just kind of playing around in my computer. As you'll see a little bit later, that some of these results are kind of strange. And my goal is just to sort of stimulate conversation around how maybe we could explore these data sets in a little more detail. So how did I do this? Okay, so I basically did a true sort of change analysis spatially. And I know the district has done this analysis as well, but I thought it would be useful to try to reproduce this on my end. But essentially, I just took for Tampa Bay the 2020 seagrass layer, the 2018 seagrass layer, and did a true change analysis, meaning that I intersected the two and looked at areas where we had sort of no change, so where the polygons were the same between the year pairs, and areas where we had seagrasses in 2020, but not in 2018. So this would be areas where they were gained, and then seagrasses where we saw them in 2018, but not in 2020. So areas where we saw a loss of seagrasses. And so again, going back to my hypothesis that if light conditions were a factor driving these changes, this is just kind of an example here showing how that might manifest in the data. Sort of looking at this intersection here, we might see that if light conditions or water clarity decreased or conditions became less favorable in 2020, we might see a shift in this polygon towards more shallow waters. and we could see, this would be where we would see a loss, and would be where we would see a gain. So that's really all I'm trying to do here, this sort of change analysis, and then relating it back to the water quality data. And so when I did this change analysis, I also looked at the gains and losses polygons and combined them with a bathymetry layer to look at what was sort of the average depth in these gains loss polygons over time to understand was there a net shift towards more shallow waters. And I summarized this at the base segment level and the seagrass management area polygons I showed you a second ago. And again, linking this back to water quality, I used essentially the EPC long-term monitoring data set, looking specifically at the 2019 data and then the 2017 water quality data, knowing that when the images for the aerial coverage maps are actually acquired, it's usually, at the end of the year for the year preceding the nominal sort of year that's assigned to each layer. So the 2020 coverage layer was actually captured in December 2019, same for 2018, going back to the 2017 late season photographs I got there. So in order to link those back to water quality, I had to sort of shift those back in time 1 year. So that's pretty straightforward there. And then when I did this change analysis, so now we're looking at some results, looking at the gained lost estimates for the different polygons. I just have some images here of different areas. So this is like Lower Tampa Bay here. Here's Hillsborough Bay. Here's Old Tampa Bay, Feather Sound, where we saw a lot of losses. And then here's kind of the southeastern edge, looking at Terracea Bay down here. And so if I just tallied up like the acreage gained losses in the different segments, it's fairly close to what the district estimates are. We all saw several presentations from Chris A. over the last couple of months. And so this is sort of confirmation that the analysis I did was more or less correct, looking at just the acreage tallies here. So again, huge losses in Old Tampa Bay, or at least the majority of the losses in Old Tampa Bay, particularly around the Feather Sound area. Other losses in other bay segments we saw as well, but again, most of that was in Old Tampa Bay, primarily in the Feather Sound area. Now, if I wanted to test this hypothesis that if I looked at like polygons gained, where we saw seagrass being gained in 2020 versus where we saw it lost in 2020, I had to relate that back to a depth estimate. So if we think that water clarity is an issue, we might see seagrasses growing in more shallow waters because they're limited at the deep edge. I had to calculate an average depth of where the seagrasses were gained and lost. And to do that, I essentially related it to a topobathy layer, as I said before, and really just sort of summarized within each polygon. I sort of clipped out the pixels from the topobathy layer and took an average depth estimate. So in this loss polygon here, I clipped out these pixels down here, took an average depth, and did the same for every single polygon, gained and losses, and then I can look at what was the average depth for all gained and all losses to see if there was a net shift towards more shallow waters. And because each of the polygons vary in size, I did this slightly differently by weighting areas or weighting polygons with different areas, with the idea being that larger polygons, maybe we should attribute more weight towards them when we're looking at an average depth change over time. So when I show the results, you'll see that this average depth is reported just as an arithmetic average, as well as a weighted average to account for polygons of different sizes being something that could have more weight towards the analysis. Before I go on, I also wanted to point out that all of these analyses are online. There's a link at the end. I have this very long sort of file here that really breaks it down. But just to show you, this is a map of all of the polygons from the 2020 to 2018 comparison overlaid on the topobathy layer here, so you can get a sense of like where the gains and losses are happening and what kind of is the depth at which we were seeing those changes. So if you want to do a deeper dive, you can totally look at this this map here and get some sense of what's going on with the data. But for like all of those thousands of polygons, if we're going to roll that up to sort of a bay-wide scale, this is the result that we see. So what we're looking at here is an estimate of the change in depth averages for all the polygons where we had seagrass lost in 2020 and all the polygons where had seagrass gained in 2020. And so we have depth down here on the x-axis. So this would be on the left, more deeper depths, on the right, more shallow depths. And then this is a cumulative distribution function just showing the distribution of the depth values for all of the polygons in the gain and loss category. And so this is for the arithmetic average. This is for the area weighted average that again takes into account the polygon sizes into those estimates. And so when I first saw this, I was kind of confused because if you look at, so here's like seagrass lost, here's the curve here, here's seagrass gained, here's the curve here. When I first looked at this, it looked as if seagrasses overall at the bay-wide scale were actually shifting towards deeper areas, which is totally like opposite of what I expected if water clarity was a factor. So I would expect, again, the seagrasses to shift towards more shallow areas because the more light limited at the deeper edge, if light conditions were a factor here. So this was kind of confusing to me. Area weighted average sort of confirmed the same thing, so sort of showing a net shift towards more deeper areas. It's not huge, but it's there. And if you break it down by base segments, same sort of overall pattern. So Old Tampa Bay, shift from shallow to more deeper areas. Similarly for Hillsborough Bay, Middle Tampa Bay, Lower Tampa Bay. Now, when you look at the area weighted changes, it's a little bit more fuzzy. We do see Middle Tampa Bay and Lower Tampa Bay sort of showing the same trends, shift towards more deeper areas. Hillsborough Bay, Old Tampa Bay, it's a little more nuanced depending at where on the CDF curves you're looking at. So a bit strange there. But there's another way we could look at this. We could actually look at the net change. So like what was the net shift instead of just looking at gain versus loss curves, we can actually take the difference of those curves and actually understand the results that way. And so when I did this, again, I did this for the bay segments as well as all the seagrass management areas. This starts to paint a picture that kind of looks a little more like water clarity conditions are driving these changes. And so again, this is showing kind of the net change between these curves, but instead of just taking the differences between that net change, I'm actually weighting that change by total area lost within each one of these polygons. And when we did that, your look, you see a net shift towards positive values. So all polygons, the net shift looks like it was moving towards more shallower water, sort of kind of look in line with the fact seagrass or water clarity was at Lower Tampa Bay. Hillsborough Bay, Lower Tampa Bay.
I just lost Marcus.
I think we all did.
Yeah, I lost him too. Maybe if he turns his camera off.
Yeah, I'm gonna Slack him.
Right, scale is a little weird because it did look like seagrasses tend to be shifting towards more deeper areas. But if we broke it down by...
Marcus, can you hear me?
Yeah.
We lost you for about a minute there. You're cutting in and out.
Let me stop sharing my screen or my camera. Yeah.
I think they're just waiting on the payoff for the seller for that one. So Wildwood.
All right.
That's my to go on mute.
Yeah, we got to mute. Okay, so yeah, I'll just back it up and I'll start with this slide here. So I think this is where I dropped off. So essentially, again, this is kind of showing the net change and depth changes by weighting the differences by total acreages lost in each polygon. And so when we looked at it this way, it did kind of look like that in Old Tampa Bay, we were seeing a net shift towards more shallow areas, which was concurrent with an expectation if water clarity is driving these changes. And sort of the opposite trend in Lower Tampa Bay, which was kind of odd, and we're not really sure what might be driving those differences as well. And breaking it down by the seagrass management areas, you kind of see similar trends. So going back to our hypothesis that is light limitation really driving the changes for 2018 versus 2020, it's kind of ambiguous, but again, this is sort of a case study if we can do this type of analysis with these broad scale, coverage polygons with the bathymetry layer to see if it's within line of these expectations. So we're not necessarily sure if this is confirmatory that light limitation is an issue, at least from how this analysis is playing out. But we can definitely look at other data sets to see if this is supported by other lines of evidence. And so I just wanted to draw your attention to the Transect data. I had some conversations with Roger Johansson last week about, does this analysis make sense? Are there other sources of data that could support this analysis? And so, if you look at the Transect monitor data, that's a much more higher resolution data set. We have information every year, and you do see kind of these shifts in different parts of the bay that sort of support what we're seeing in this broad scale analysis with the coverage polygons. So this is one transect for Old Tampa Bay. You kind of see the seagrasses, looking at the 2017 versus 2019 late season when the images were acquired by the district. You do see in this transect, looks like it might be growing more shallow. Here's one for Lower Tampa Bay. You kind of squint, not really sure, but it does look like it might be growing slightly deeper in that part of the bay. Not really sure, but again, just sort of making you guys kind of think about other data sets that we can use to support this analysis. Now, the question is, what's happening with actual water quality measurements? Do we see a change in water clarity conditions that, might make us think that this is actually driving the changes in the seagrasses in Tampa Bay from 2018 to 2020? And so what this is showing you is for major bay segments, Old Tampa Bay, Hillsborough Bay, Middle Tampa Bay, Lower Tampa Bay, looking at Secchi in the top row, turbidity middle row, color in the bottom row. Long-term record here, but highlighting the 2018 and 2020 years, or rather 2017, 2019 years for the water quality, what the changes were between those year pairs. And if we pull out those 2 year pairs for all these measurements by bay segment, we can get a picture of what those changes have been as a means to maybe driving those changes in seagrasses. And so Secchi, it's a little ambiguous. Some parts of the bay, it looks like Secchi measurements are getting, less, so light attenuation is increasing in Middle Tampa Bay, Lower Tampa Bay. Turbidity is a little odd as well. Old Tampa Bay, Hillsborough Bay looks like turbidity might be going down. But bay-wide, what's interesting is water color is actually increasing in all bay segments from 2017 to 2019. And this is maybe creating conditions that could lead to possible light limitation in seagrasses. And so, not just looking at Secchi disk depth, but other variables as well that could explain these changes. And this is just showing percent changes by seagrass management areas for the different water quality measurements. And this is also kind of tricky because the EPC measurements are more in the middle parts of the different bay segments. So it's kind of difficult to relate these back to the management area polygons. But again, just sort of looking at spatially how these changes play out.
Marcus, you have a question from Chris Kaufman about whether or not you can query like the Transect data for epiphyte coverage and drift algae to see if that's maybe part of what's driving the changes.
Now I haven't looked at the Transect data. The long-term record of the drift algae is there in the Transect data, but it's kind of it can be fairly generic at times, so it's difficult to really tease that out in terms of looking at, maybe biomass coverage or abundance estimates. But I have not looked at that, and that could be another line of evidence to sort of understand what these changes or what's driving these changes.
Yeah, I think in the last couple of years, the last year plus maybe, we've started to improve the drift algae on an overall basis for the system for that analysis. But I don't think going back beyond that, you can make really good overall statements. And then on the epiphyte, it would have to be sort of it would definitely be qualitative rather than quantitative, because the epiphyte loads are identified as clean, light, heavy, moderate, heavy.
Yeah.
But definitely, thanks, Chris. Definitely recognizing that those can impact seagrass growth.
Yeah, and haven't looked at that. Again, this is, totally me just playing in my own little sandbox. And those types of questions are great because I can start thinking about how I can maybe do a more comprehensive assessment to look at other pieces of evidence here. Yeah, and so I initially intended to sort of compare those percent changes in Secchi, turbidity, and color to the net changes and seagrass depth, and maybe trying to look at correspondences between the two. And I'm not going to show it here because it was messy and it really sort of confused me. So, and that could just be a limitation of just the scale of analysis we're looking at here. And not necessarily giving up on, obviously water clarity we know is the main thing that's driving seagrasses, just generally speaking, but looking at, just these 2-year pairs of data and this scale of analysis, it might be, other factors that are limiting our ability to really understand what's driving these changes. So that's why I'm not going to show that here. But overall, if you do look at the trends, water clarity conditions are changing in the bay, and that I think is something generally to be concerned about. And yeah, so I wanted to finish my last couple of slides just focusing on this bigger picture. Whether or not this analysis is informative or helpful, I can certainly, we can discuss the merits of that, but by and large, if you step back and look at bay-wide trends, not just in Tampa Bay, but at the region as a whole, I think there are some things that we need to think about. And this is just looking at old Tampa Bay. For the last couple of years, we've sort of bounced above our threshold for chlorophyll A. We've been doing good for light attenuation, but if you look at the trend over the last 5 years or so, you do see that it is creeping up and up and up, suggesting that water clarity, that is a trend that we don't want to see. And it's something that I think is going to be more of an issue in the next couple of years in terms of what where we focus our efforts towards, recovery efforts in that part of the bay. Other issue, non-point sources. This is for Old Tampa Bay as well. But in terms of what's driving those changes, I think we need to think about sources of nitrogen into that part of the bay. These are estimates from Janicki Environmental. Obviously we've done a good job of controlling sort of the main point sources, industrial point sources, domestic point sources over the years. That's just been kind of our recovery story over the last couple of decades. But looking at the trend for non-point sources, you do see that kind of increasing over time. And I think that is where we need to sort of focus our thinking around other aspects of sources of nutrients into the bay, given that this trend is increasing. Again, also focusing on Old Tampa Bay, there's also certain times of the year and certain events that I think we need to think about in terms of when we see elevated loads entering the system. This is actually a slide from a presentation I gave last year to the Nitrogen Management Consortium, but looking at basically chlorophyll trends by different seasonal parts of the year: January, February, March, April, May, June, July, August, September, October, November, December, and then looking at the long-term trends by season in chlorophyll and how those kind of track hydrologic loads into the system. So basically times when it's rainy, we see large influxes of nutrients to Old Tampa Bay, and those tend to correspond to these, anomalous or sort of high seasonal values in chlorophyll A. So it's not just non-point sources of pollution that we think are maybe a larger issue here, but certain times of the year when we see those huge loads coming into the bay, and what that means for driving changes in water quality and how that translates to potential effects on seagrasses. And then also the bay as a whole, this is a slide from that same presentation. Interestingly enough, salinity, these are just annual averages, has seen a net decrease over time in the last 10 years or so. So Old Tampa Bay, Hillsborough Bay, Middle and Lower Tampa Bay, looking at average salinity from the EPC estimates, there's just been a reduction over time. And I think that's a broader trend we see regionally, not just in Tampa Bay, that could change obviously conditions in the bay, but could affect what we see with the seagrass community, because we know that some species are more tolerant to salinity than others, and that could shift species compositions towards more salinity tolerant species like Thalassia, for example. So bigger picture, things to think about. Take-home messages, obviously, as I said before, this polygon change estimate, I think it's a useful exercise, but definitely a first cut of this type of analysis. And my goal today was just, again, to stimulate conversation around how we might think about leveraging different sources of data, looking at different hypotheses and trying to challenge the data in different ways to address those questions. Again, with the specific idea of looking at 2018 versus 2020, but looking at these broader trends that we see as a whole, not just in Tampa Bay, but other locations. So light attenuation is still going to be very much an issue for understanding changes in seagrasses in Tampa Bay. But what's driving those changes is the non-point source loads, other factors related to, regional patterns and precipitation, what that means for salinity. All these things are important to think about in the broader context. So I just, want to pose a question of what will the future of the bay look like? Obviously we don't know at this point, but I think we can look at these data sets and be able to sort of understand changes we're seeing now to sort of get an idea of what's going to happen in the future. And obviously there's other data sets we can use to sort of understand those changes. The Transect data came up. That's certainly one way to look at, but we have, some talks later today looking at, for example, remote sensing products as another sort of lens to understand those changes. So yeah, lots of big questions, maybe some small answers here, but again, just trying to understand what we see this year and scaling that up to other changes. So that's all I've got. I've probably talked way too long as usual, but here are some links to some resources that might be helpful. This is that full analysis page I showed earlier with this map and all the code to do a lot of the analyses we saw in my presentation here. Here's the dashboard that I linked earlier that looks at the change estimates by coverage. And just want to throw out there too, also the Transect monitoring dashboard is still out there. I think it's a great resource to do this deeper dive, looking at changes at the Transect level scale as another resource there. So yeah, I see a lot of questions popping up in the chat. So if anybody has any questions, feel free to unmute and we can hopefully have a few minutes to answer some questions. Thanks.
Yeah, we've got a couple of minutes. Thanks, Marcus. That was a really good overview of the way that we're able to use, the new analytical techniques to look at the projects that we've been working on for years, if not decades. Going back, I think Chris had the Chris Kaufman, I think, was answered during the chat. But Chris A, you got a couple of things in here. Do you want to expound on them?
Yeah. So, first of all, this is really great stuff, and it does encourage us to kind of start thinking about the cause and effect relationship. So I appreciate everything that Marcus, you've done with putting all these data together. One of the things I think that would be interesting is to look at the seasonal water quality. So look at chlorophyll during the growing season, water clarity during the growing season, because it tends to be, as everybody knows, there's a seasonal signal there that might help tease some of those anomalous readings or maybe change that story a little bit to where it may lend a little bit more credibility to the hypothesis that we are actually seeing lower light conditions. And again, too, just to reiterate, and why the Transect data is really important. When we look at aerial coverage of seagrass, we're looking at spatial distribution, but we're not looking at quality. So you could have grass growing in deeper areas, but they may be under some sublethal stress that just doesn't get picked up in the signature. So that's just something I just wanted to point out. But I think, yeah, if you look at the water quality data, like at the end of the growing season, might be a different picture. The other comment I had was just about, and I like Dave's analogy of a cage match. So like celebrity death match between Caulerpa proliferans and Haliduli, who's going to win if you increase the nutrient pool in the water column? And I think, it would be Caulerpa because it's getting most of its nutrients from the water column, as opposed to Haliduli, which is, getting most of their nutrients from the sediment. So I think competition may be driving some of this, and that might not be showing up in the water quality or the water clarity data. So that's just another thing to consider, because a lot of the trends that we see in terms of the overall numbers in Tampa Bay are being driven by Old Tampa Bay. And we know that in Old Tampa Bay, we've seen almost a 1-for-1 swap from Haliduli dominant, Haliduli rhytii to Caulerpa proliferans. So those are just some of the observations I had. But yeah, really, really cool stuff.
Yeah, thanks, Chris. And we've definitely seen that shift too. I mean, I think last.
No, I'm sorry, that was me. I was trying to move stuff around, and the right-click function did not work the way I thought it did. Sorry for muting you all.
Yeah, no, I was going to say, yeah, Chris, the species shift is something that we've definitely discussed in the past. Definitely the feather sound transect.
So I apologize. I am on 2 meetings, and so my client just started addressing this other topic. So I wanted to hear that. I tried to mute you. I didn't understand I was muting everyone, because on that meeting I can mute someone and still hear you guys with.
I'm not sure he's talking to us.
Yeah, I don't know if that was.
Yeah, we need to mute that.
Yeah, wrong mute.
Yeah. Well, I don't think I'm muted, so I'm just going to keep talking.
No, I hear you.
Yeah, so the Caulerpa shift, definitely of concern. Feather sound is one place that we've seen that happen. And you don't see that in the coverage maps. And, that could, be obscured, but definitely the Transect data is an alternate source of information to try to understand those changes. And my analysis is very coarse. The water quality conditions, definitely something to think about. I was just basically looking at 1-to-1, 2017, 2019, what the change was. But, there's sort of cumulative conditions that, if you have, poor water clarity persisting for a while, if there may be a threshold there, that could be driving a change in seagrasses as well. And that definitely wasn't a way I sort of looked at the data, and certainly the seasonal patterns as well, growing season conditions only, or what was preceding the growing season. Definitely certain ways you could slice and dice the data to get at this question in more detail. And I'm personally a little skeptical too about how I did the polygon analysis in terms of like, it'd be great to track like one bed between 2 years, but the way I've done it, it's basically like an aggregate of all beds by segment, and it's can be challenging to try to understand gains losses at that scale. And I was hoping that it might sort of come out in the wash, this sort of net signal looking at that large scale. And I think some places it did, but I don't know if it's you can sort of challenge the data a little bit more by at the bed scale, because I think you could do some more pointed analyses. But anyway, this is sort of phase 1 of a larger project, perhaps, with these types of data.
Yeah, and Marcus, just another thing to keep in mind too, for the benefit of the group too. And you mentioned this during your presentation that the aerial photography is collected during the winter season, so you're at a minima of seagrass expansion. If we were collecting imagery in October or September, that signal again might be different because of that.
Yeah.
Okay, good discussion. Thanks, y'all. Need to move on to Chuck Jacoby. I just found out he's got another meeting coming up shortly, so we'll save some of there were several more questions that are coming along. We'll leave those for the end if we've got time. I will.
Could I just apologize? I was trying to multitask and unmuted my collective mic to everyone, so I apologize for that.
No worries, Nate.
So our next presentation is from Chuck Jacoby of the St. John's Water Management District, and he is going to be discussing some of the Indian River Lagoon seagrass issues.
Hang on one tick. Here we go. Thanks, Gary. So people seeing the PowerPoint at this point?
Looking good.
Okay. So as Gary said, I'm from the St. John's River Water Management District. We deal in part in my group with the Indian River Lagoon. And I was asked to come and kind of give you a little potted history of what's been going on there. So hang on. Okay, got it. So we're going to go with the bottom line up front approach, so that folks, if they want to, can watch the little animation and then go get themselves a cup of coffee. So this is overlays of maps, actually run from 43 to 2019. What you want to keep an eye on here, this Banana River Lagoon is around the 2011 mark when things really changed in the lagoon, not only in Banana River, but elsewhere as well. So up to 2007 was our sort of peak year for acreage or hectares. And then after that, let's see, stalled. There we go. So then 11 came and we lost a lot of seagrass. 13 and 15, we saw a little bit of an uptick. 17 through 19 down, and 21 maps are in preparation at the moment. So let's take a little closer look. This graph on the left is seagrass extent in acres. That's the green bars. And on the right is mean transect length, in this case in feet. And what we used these for is because the transect length and the acreage correlate pretty well. So we're able to look in between the mapping years to see what's going on. And this is the initial part of the tracking that we've done. So sort of running from 92 on, as it were. And the seagrasses were expanding, which everybody was pleased with. We passed the 1943 extent, which may indicate that in 1943 there already were impacts on the seagrasses. And on the bottom there, I've tried to sort of put some events in there to give you an idea that it was both good management, discharges, the point source, the major point source discharges into the lagoon went away in the 90s. So we really haven't had that since then. Some of the water control districts, water was diverted back into the St. John's Basin because the canals had been dug to move water to the lagoon. But we also had good luck. So we had a fairly dry period of time. And when we did have rain, like in 2004 with hurricanes and 2008 with Tropical Storm Faye, it was a lot of rain all at once. And that seemed to, in a sense, reset the lagoon almost. So that's, again, just a little more detail, sort of changed around that 2011 mark. So here again, you have the aerial extent, the transect length in meters this time, and also the yellow is the percent cover on the right-hand axis as well. So we had, increasing transect length and acreage. Then we had the drop with 2011. We had that uptick, which is the orange arrow, and then the drop ever since. And if you look at the cover was hovering around 20%, and then it started to drop below 20%. And in fact, some of the analyses we've done now say that that sort of 20% average that we saw, that below that, the rapidity with which seagrasses decreased in cover increases. So that 20% is a kind of a threshold, and below that, for every month of bad light, you tend to lose more seagrass. And in fact, it was bad light that we think drove all of this. So here you see 3 different parts of the system with the percent of incident radiation at 0.9 meters shown on the graphs over time. And the arrows point to the places where, the excursions below this 30%. And that's sort of where we think things begin to be stressful for most of the grasses in the lagoon. Those excursions became both more extreme, lower light, and longer lasting. And so what was causing all of that? Well, we had a series of phytoplankton blooms. These are satellite images with modeled chlorophyll concentrations, the hotter colors being higher concentrations. And as you can see from here, since that 2011 period, we've had a series of blooms that were both intense in terms of how much chlorophyll there was, fairly widespread, and that seems to be driving the changes in the light regime. Okay, that went the wrong way. There we go. Okay. Sorry. Okay, so we saw the changes in seagrass. We've had, this sort of situation for a long enough period of time and a couple of decreases in their extent and cover. We're actually beginning to see some flow-on effects, we think, in some of the other parts of the system. So these are data from the Fisheries Independent Monitoring that FWC undertakes. The code goby, which is the graph in the upper left there. Code gobies are obviously really tied with seagrasses, and so you kind of see their abundances tracking the dip when we first lost seagrasses and the uptick and then the dip again. But we're even starting to see it in the juvenile snook, which is the lower right-hand graph, where their abundances, again, seem to show that pattern. It's a lot noisier because obviously recruitment puts some noise into that system. There we go. But we're also beginning to see flow-on effects even more broadly. So there was a 2012-13 UME for manatees. And that one now, it appears there was a change because of the change in diet where they shifted from seagrass to drift macroalgae. Some of the manatees couldn't cope. They had a gut problem, a Clostridium, produced toxins, and it gave them a kind of a toxic shock syndrome, and they died. The 2013 dolphin UME really hasn't been closed out yet, but it occurred right around that same period of time. We've also seen fish kills when the phytoplankton blooms crash and the DO goes down. And then recently we had the most recent manatee UME, which appears to be the first one that's really due to the lack of food. So the animals are emaciated and in really bad condition. So what do we think happened to cause all of this change? Well, a bunch of folks got together. We had some funding from the district and from some other places, and I steal most of their results and put them together. And in my head, this is the kind of model that helps me organize my thoughts. It's Duarte's eutrophication progression scheme, where you're looking at a system that has loads of nutrients pounding on it. However, the system doesn't respond immediately. It doesn't collapse immediately because there are parts in the system that allow that system to cope with that increased loads of nutrients. Eventually, either the loads are too high or the shock absorbers in the system, one of or more of them give way, and you get enhanced growth of phytoplankton. That's where the lagoon went, and or macroalgae. Increased shading, increased benthic respiration typically, which means sulfide production in the sediments, and the seagrasses kind of get squeezed with low light and sulfide toxicity. And so there's a fairly rapid loss of seagrasses. So if we try to delve into what if anything was going on in the lagoon at the time of that 2011 bloom, We see that the loads, in fact, were not particularly high. So that's total nitrogen in pounds per day in the upper left for 3 different parts of the system, total phosphorus in the lower right. And the period of interest is marked by that bracket there. And that's from the modeled loads. So, that's our best estimate of overall loads. And in fact, if you look at the rainfall, which drives those loads, obviously, the rainfall was not particularly high either. So we're probably looking for something in the system rather than just external loading. We did see in the system a change. Particularly noticeable was the change in the nitrogen to phosphorus ratio. So the lagoon shifted. There was more phosphorus relative to the nitrogen around that 2010-11 mark. And if we look at how that matches up with chlorophyll, so in the upper left, it's phosphorus concentrations in the water column. On the left-hand axis, that's the orange, and then the green is chlorophyll, which is on the right-hand axis. You can see that as the phosphorus levels rose, the chlorophyll levels also rose. And we've known for quite a period of time that the lagoon has a kind of a gradient of phosphorus increasing as you go south. So this northern part, it's not maybe a surprise that increased phosphorus drove increased chlorophyll. In the lower right, that's nitrogen on the left-hand axis, the blue, and again, chlorophyll in the green. You don't see quite the correspondence, but you also have to take into account the fact that nitrogen concentrations were fairly high already. So what might have caused these shifts that we're seeing? Well, there were some somewhat unusual events in conjunction with that 2010-11 period. So a couple of pretty cold winters and a couple of fairly warm summers. And that in combination with a bunch of other things appears to be part of what moved this system. One of the things we saw in January 2010 was a cold kill of fish. So up in the area around the Kennedy Space Center, the NASA contractors collected up 400 tons of fish. And the estimates are there for what that might mean in terms of nitrogen and phosphorus getting into the system. And they estimated that, given the area they covered, you could probably triple it for the area of the fish kill. So there's an internal source. Another internal source that we've been dealing with for a long time, as it were, is what has become known as muck, which is fine sediments. It's really only got about 10% organic matter, but that's higher than the typical sandier sediments in the lagoon. And this sort of accumulates in the low spots where they've dredged material. And in the intracoastal waterway, that's the map on the right shows the yellow is the muck, and you can see it following the intracoastal waterway there. And what's been found is that there's a pretty strong temperature effect on how nutrients, nitrogen, and phosphorus flux from this sediment. So as temperature is up, like we saw for the couple of warm summers, the flux is going to be up. There's also a pH effect. So the flux goes up as the pH increases, and the pH increases as the phytoplankton draw the CO2 down. So a couple of different things might be driving the flux from the muck. Unfortunately, we don't have really great spatial temporal resolution on flux from muck. It's not a simple thing to track. And we're also finding out more now that we may have the bottom water becoming a little decoupled from the surface water. I mean, the lagoon is shallow. For those who don't know, it's maybe a little over a meter deep on average. But it turns out that it is possible for there to be low DO at the bottom and reasonably high DO where it's mostly measured. So there's another internal source. Macroalgae, drifting macroalgae is something that we see in the lagoon. This is an image showing you what it can look like at times. And again, this potentially is another way to affect the cycling of nutrients in the lagoon. Drift is typically a bycatch measurement from different kinds of sampling. In this particular instance, it's seining that's done by the Fisheries Independent Monitoring Group, and the mean biomass of drift algae is the y-axis over time. But the arrows point to a couple of places, that 2010-11 slot and then the 16 slot where we again saw a large phytoplankton bloom. And what we were seeing there is less drift algae in general, so less competition, as it were, for the nutrients that might be available. We have some tank experiments that indicate that the drift algae don't grow as well when it's warm or when it's cold. We didn't really see mortality. We just saw kind of a lack of growth, but that may have been enough to shift the cycling. And in fact, now that we have a couple of events, the 11 and the sort of 16, we can look at the overall distribution of carbon, the estimates of carbon distribution among those three key primary producers. So you can see that the green shape, that's the seagrass biomass. Then that's in the upper right, that's on its own there, and the arrows show you where it dipped. And then we add the drift, which again is on the same axis, and that's the brown line. And then if you add the phytoplankton, it kind of fills in the gaps. So the system has moved from a system where the carbon, nitrogen, and phosphorus is, sort of bound by seagrasses, which turn over fairly slowly, and drift, which is kind of in the middle, and it's much more flashy now, with the phytoplankton being the driver of the system. But it's not just the quantity of things that have changed, it's also the quality. So what we've seen is a shift from diatoms and dinoflagellates that you can see on the left hand with the boxes and the little pictures, and some of the larger cyanobacteria to picocyanobacteria and small nano chlorophytes and brown tide, which is an nanoplankton as well. And just recently, one of the blooms was a different kind of small cyanobacteria that we hadn't seen before again. And it appears from what we know about these that we've learned, some of which are still yet to be identified completely, is that they are worse actors than we were used to. So the diatoms and dinoflagellates were a little less disruptive of the system. And some of the reasons for that are listed here. These new things are smaller, or they turn over faster. Brown tide, for example, has a resting stage. There is some evidence that some of these newer players can use organic nitrogen and phosphorus. They don't need the microbial loop to strip those elements off the carbon. The picocyanos may actually fix nitrogen at times. Again, there's a little bit of evidence of that. But we also know that the top-down control is probably not as strong as it used to be. Microzooplankton may do all right on some of these things. Mesozooplankton, they don't seem to do well, particularly at high densities. Brown tide is not preferred by much of anything. It produces a mucus coating that seems to inhibit a lot of the grazers. So what does all this stuff add up to? Well, still it's nutrients and leading to chlorophyll. That's probably the key issue, and that is being addressed in the lagoon. So these are graphs showing you the loads of total nitrogen in the upper left and total phosphorus in the lower right. And the bars go from the darker bar, which is where the loads were, to the bar, which the medium colored bar, which is the load in 2019, and then the target. And so you can see we're somewhere between 21 and 65% of the way to the target. And again, it's important to remember that this is not point sources. The point sources were gone before this whole process even started. So we're dealing with the non-point sources, which we just heard a little bit about in the Tampa Bay, and the fact that they can have a pretty important driving effect. And so overall, it may not be quite as simple as Dave Tamasco's statement about you don't want to be like the IRL, because it's the parts of the system, and the IRL in general has a high residence time. So it's both special in the sense that you get certain kinds of habitats and certain kinds of ecology there, but it's also really sensitive. So the residence time runs from months to years in parts of the lagoon. And that may be a big part of what's going on. It just can't cope with the loads. Okay, so some other take homes. Probably need to look beyond loading to cycling to understand how nutrients function in the system. Probably need to look beyond chlorophyll to see who's creating that chlorophyll when it comes to the phytoplankton, because again, we're seeing some bad actors, as it were. Also, the shift in the system was probably something like a perfect storm with a conjunction of events that altered the way nutrients operated in the system, and it's sort of become a bit of a new norm, which to me means that you might, when you consider the management of things, you might want to think about events a little more than we tend to kind of leave a little elbow room in the system, in the way you're approaching management. And I think, you know, one of the real take homes for most people will be that this shift is going to be a challenge to restoration probably. So we're going to be looking probably at some sort of lag. We may have to push back a little harder on the loading to get the system to come back to where it was. And obviously we came up with a whole bunch of new questions, which we'll hope to investigate as we go forward. Internal loads, you know, including submarine groundwater, which again, we get in the lagoon, but we really probably don't have the best handle on exactly what that load looks like. How drift algae plays in the system. Again, nobody focuses on drift algae. It's painful to sample, but there also may be a management tool there. You know, you may be able to harvest it and remove some nitrogen and phosphorus. Phytoplankton probably need to understand a little bit more about how they interact with each other and their life cycles. That definitely need to know more about how they use nitrogen and phosphorus. Filter feeders, you know, we had clams in the lagoon. They went away sort of in the 80s. The clam populations seem to have been hammered too heavily by fishing. So bringing back filter feeders may be an important step. And then again, if we get the water quality to a stage where it's suitable, I don't think people are going to be patient enough to wait for seagrasses to come back from the 58% loss of acreage that we've seen on their own. So we need to maybe prepare to help the system get back on its feet. And that involves a whole range of things, including just the simple thing of where do you get the supply of seagrass now that there's so little in the system itself. And I'm happy to try to answer any questions.
Thank you, Chuck. Definitely, you know, sobering news from the St. John's. I think some of this we'd seen before, but appreciate you putting it in that perspective, and especially thinking about, I think, echoing what Chris has in the chat about, you know, keeping room for those episodic events, understanding that they're part and parcel of the system. Do you have time for a couple of questions?
Sure.
Okay. I threw down a quick one. One of the slides you showed average lengths for the transects of the Banana River. Do they all start out the same? How do you get that average?
No, the transect that was for the whole lagoon, and so that's just the average transect length over the whole lagoon. Different parts of the system sort of responded differently for a range of reasons. The bathymetry, for example, Mosquito Lagoon is broad, shallow shelves, and so that you don't really work at 0.9 meters. You know, it's shallower than that. But also the timing of the blooms. So, the places that got hit the hardest, the bloom came on as the growing season started. If it came on later, because the blooms, all those parts of the system are kind of connected by canals. And so eventually blooms can spread, but the timing makes a difference. So a range of different things. But the graph I showed you is just the overall average for the lagoon.
Okay, thank you. Chris A., do you had a question about the mid-solids?
No, hey Chuck. Yeah, my question was about sea surface temperature. I was just curious. I think you already covered it, so it's kind of, you answered the question, but I was just curious if, like, over time, and I'm talking like long-term trends, maybe not, over the last few years, but there may, have you seen an increase in sea surface temperature associated with just the general warming of the climate? And perhaps could that be, contributing to this increase in these algal blooms? Not so much that, you kind of reach a tipping point where now you're in a regime where your growing season is longer, and so these phytoplankton have the ability to reproduce longer than they ever have before.
Yeah, I mean, probably we do see a slight trend in, overall sea surface temperature. Certainly we've had some warm winters. I think if you looked at the satellite imagery, some of those were from February when you had, a fairly major bloom, which is a little bit late in the season, as you pointed out. So those warm winters, have not been helping us. We could use a good cold winter to just knock things on the head and, settle the system down a little bit. So, whether that's, how much of a trend that is a little hard to tell potentially, but.
Yeah, well, I'll tell you from anecdotal observation on this side of the state, what we've noticed is not necessarily that things are getting warmer, but they're getting less cold. So you hit on it with, the winters have been, exceptionally warm over the last decade. I can't remember the last time we had a hard freeze on this side of the state, but it's been a long time. And so I think, the phenomenon that we're seeing, which is hard to tease out sometimes when you're just looking at either spot measurements or you're looking at, average temperatures, is that these systems are potentially, anyway, getting less cold. And so that may be really where the signal is hiding, so to speak, in the data.
Yep, and for ours with the drift algae, if it tends to get warmer in the summers as well, then that could lead to some challenges because you don't have the drift algae hanging on as long, so you don't have that competition for the nutrients through that initial growing season.
And I wonder too, like with Mosquito Lagoon and perhaps Banana River, I know like with Mosquito Lagoon, that's hypersaline. If you're increasing temperatures, presumably you're going to increase the evaporation rate. So potentially, do you think salinity might be driving some of this? I mean, are you seeing any long-term trends in salinity, especially in those more hypersaline areas?
I would say, probably not a clear trend at this point, but certainly hypersalinity around that 2011, there was quite saline, particularly like you say in Mosquito Lagoon. And the first brown tide was 12. The salinities were really high, but the second brown tide, the salinities were brackish. So there is some literature says, brown tide wins in high salinity, but brown tide wins other times as well. So, it's not that clear, but I mean, all of those things, as salinity, temperature, and light, all those things interact to stress seagrasses. So, if you get a combination of high salinity and, really high temperatures and low light, seagrasses are probably really struggling. If they have light, they probably can cope with the other two reasonably well. But if they, if you take that light away, then, it's a triple threat. So.
Okay, thank you so much, Chuck. Again, really appreciate the presentation. I think we're going to move on. Again, there's several questions that we're leaving unanswered right now. Hopefully, we can either get back to them at the end of the discussion, or, Chuck, if it's okay, I'll share your contact information.
Yeah, sure. And if you want, if you send them an email, I can probably, type back some answers as I go.
I'll try to follow up with some of the questions that are in the chat and see what we can do.
Yeah, sorry to eat and run, as they say, but
no worries. Much appreciated. Take care.
Okay.
Next, we have Barbara Welsh from South Florida Water Management District, going to talk about seagrass monitoring and mapping in the Caloosahatchee Estuary.
Gary, did you get my presentation that you can?
I did. Would you like me to share it?
Yes, because I can't control it. I'm on my work computer and I'm on my home computer and I couldn't get it over there. So if you could control that, that would be great.
Can everybody see my screen?
So
Yes.
I'll just tell you next
Yes.
slide, go.
Okay, that sounds fine.
Okay, so I'm Barbara Welch with the South Florida Water Management District in the Coastal Ecosystem section. Nathan put together this presentation, but at the last minute, something came up and he wasn't able to make it. So bear with me. I just found this out like late yesterday afternoon. So Nathan oversees the monitoring program and I oversee the mapping program. Next slide. So the data presented today establishes the pre-SERP project baseline conditions for SAV and the CRE. The project supports the Recover program for long-term monitoring trends of ecological indicator species to assess SERP. The monitoring program is the Northern Estuaries SAV Ecosystem Assessment Project. It quantifies relationships between environmental variables. And the current method started in 2018. We actually go back to 2008. We had new staff come on, and in 2018, we changed our monitoring methods. Next slide. So we monitor SAV on two scales, the aerial mapping landscape scale and the fixed transect patch scale, which is the monitoring the transects for SAV percent cover, canopy height, shoot density by species, and to detect changes among SAV species relevant to timescales. Next slide. So our Seagrass aerial mapping area covers southern Charlotte Harbor at Boca Grande, all the way south to southern Estero Bay at Wiggins Pass. Next slide. So the last imagery was collected between December and January, December 2013 and January 2014. There were 370 tiles and 330 square miles. You can see there's a lot of inlets there, which caused problems in water quality turbidity. So it takes a lot of monitoring from outside agencies to help us coordinate the perfect day to get those flights done. Next slide. So our mapping area is divided into 5 segments: the Matlacha Pass, Pine Island Sound, San Carlos Bay, the tidal Caloosahatchee, and Estero Bay. Next slide. So right now, this is the most current mapping that we had done. Our mapping is done every 4 to 6 years on the West Coast. So currently we had mapping is currently being done, but it's not quite finished yet. So in 2014, we had 13,000 patchy areas, and dense continuous, we had 35,132 acres, for a total of 48,647 acres mapped for these 5 segments. There were 317 field verification points, and half-acre minimum mapping unit. And the change analysis was done between 2008 and 2014. Next slide. So the total gains in the 5 segments was 5,197 acres, and the total losses was 1,704 acres between 2008 and 2014. Next slide. So here's the breakdown of the different segments from 2008 to 2014. As you can see, we gained 3,494 acres, which was a plus 7.7 change from 2008 to 2014. Next slide. So as I mentioned, our 2021 West Coast seagrass mapping was flown in March, April of 2021. We started last year, but apparently we had a lot of turbidity issues and the weather wasn't working for us to get the mapping done in that time frame. So our contractor got it flown between March and April, and we are currently looking at the draft map and hope to have a final map with the change analysis expected in January next year. Next slide. So the fixed transect methods, there's 3 sites that we monitor in the Caloosahatchee River and Estuary. They're monitored March through November and since June of 2018 when the methods changed. At each site, SAV, macroalgae species, and percent cover, blade counts, and canopy height are taken, along with water quality data and light profiles. So this is a salinity gradient from our CRE2 site all the way to the CRE8 site. The upper estuary is mostly freshwater, and our species are Vallisneria and Rupia. And in the middle estuary, we have Rupia and Haliduli rhytii. And in the lower estuary, CRE8, we have marine seagrasses, Haliduli rhytii, Thalassia, and we also have Syringodium, Decipiens, and Engelmanii in these areas. Transects are set up at each site in the CRE. 11 points along each transect are monitoring using a half-meter squared quadrat for a percent cover score, canopy height, and blade number are measured. Brombraceae are converted to percent cover according to the Van der Merel 2007. This conversion allows percent cover for Brombraceae score of 5 to up to 140% as the scale accounts for overlapping canopy. Next slide. So average percent cover for CRE2 up at the in the freshwater area, average percent cover is 0.6, and the CRE5 percent cover average is 7.3, and CRE8 average percent cover is 28.3. So seagrasses do better, of course, in the lower estuary. So to compare similar months among years, mean SAV percent cover values from March through July were pooled for comparison among the 3 years, 2019 to 2021. Within this range of months, 2019 SAV percent cover was lowest at 5.47%, while 2020 and 2021 had similar mean SAV percent covers of 10.5 and 10.8, respectively. SAV mean percent cover at CRE varied significantly by year and was significantly lower in 2019 than in either 20 or 21. Mean seagrass percent cover in 20 and 21 did not significantly differ. So CRE2 site, next slide. CRE2 water conditions are dark water with high CDOM and low salinities of 8 to 10. The typical SAV found there again are Rupia and Vallisneria. Next slide. So the X indicates no SAV. The dashed line is the overall mean. There's a very low mean percent cover of 0.6. SAV was absent from most quads. There's no significant difference over time. And since 2018, there has only been Rupia until most recent surveys in September and October when Vallisneria occurred in 2 of 33 quads. Next slide. The middle estuary CRE5 site, salinity varies between 0 and 35, and mostly found is the shoal grass at this site. Next slide. The average percent cover is 7.3. And to compare similar months among years, mean SAV percent cover values from March through September were pooled for comparisons among years 2019 to 2021. Within this range of months, 2019 SAV percent cover was the lowest at 5.47, while 2021 had similar mean SAV percent covers of 10.5 and 10.8 respectively. Next slide. The lower estuary salinity is typically between 20 and 35. Shoal grass is the foundational species tolerating variable salinity and disturbances. Turtle grass favors the marine salinity waters with low turbidity and is the apex species if no disturbances, higher salinities, and light are present. As mentioned before, other species found here are also Syringodium and Engelmanii. Next slide. The overall average percent cover for the CRE8 site is 28.0. When the percent cover of SAV for months March through July were pooled for comparison among years 2019 to 21, there was no significant difference identified among years. The Braun-Blake scores were converted to percent cover according to the Vander Merrill 2007. This conversion allowed for the percent cover for Braun-Blake score of 5 to be over 100%, as the scale accounts for the canopy complexity and overlap. Next slide. As we continue to add new data, our perspective on the range and variability of SAV resources in the CRE will expand and better represent normal long-term conditions, which will help discriminate among changes due to the implementation of SERP projects and the effects from other drivers of significant changes in SAV, such as the hurricanes, storm events, drought, and sea level rise. As this data set matures and we gain a better understanding of how SAV metrics change in response to environmental conditions, we can assess seagrass species growth and survivorship in response to changes in temperature, flow, and availability of light and depth. Any questions?
Thank you, Barbara. Much appreciated. Always fun to try to do somebody else's presentation at the last minute.
Yeah, really.
with construction going on all around you.
You can hear that, huh? Sorry.
Just a little bit, no worries. Can you share the was it you said Vander Merrill reference from 2007 conversion of the Bromblon K-scores? Either in the chat or email it to me, because I don't think I've seen that one before, and
Yeah,
I'd
can
be
do,
interested in looking at it.
can do. We
Any
also
other
do,
questions?
Coastal also does monitoring on the Indian River Lagoon and the St. Lucie River. We have a few sites there, or St. Lucie River and Estuary, but today we just presented on the Caloosahatchee.
Thank you. Any other questions for Barbara right now?
Hey, Gary, if you could pass along that reference to the group, that'd be great.
Absolutely.
Thanks. And thank you, Barbara.
Uh-huh.
Okay. Our next presenter is Nicole Yadavia from Coastal Heartland National Estuary Program. Nicole, I'll go ahead and give you the power if that's okay.
That sounds great. I just sent a screen sharing request. Okay,
Very
great.
good.
It looks like everybody
We
can see
it.
that.
Yep.
Okay. So I just wanted to, oops, let me just get this out of the way here. Okay. Thank you all for having me again. My name is Nicole Yadavia, and I serve as Research and Outreach Manager for the Coastal and Heartland National Estuary Partnership, or CHNEP. The data I will be presenting today was compiled on behalf of all of the scientists, resource managers, and community members in this region who have been involved in collecting this information by myself at CHNEP and Betty Stogler from Sea Grants. I will be giving a brief overview of the state of our knowledge as they pertain to seagrass and water in our region, stretching from Donna Bay and Venice through Charlotte Harbor all the way through Estero Bay. And again, I just wanted to acknowledge all the individuals and entities for contributing to the ongoing body of data that we have up to this point and their continued efforts to better understand waterways and how to improve them in our region. So I just wanted to share a quick map for those of you not as familiar with our region, maybe from the other coast. So again, CHNEP area covers multiple estuaries ranging from Donna and Roberts Bay, a little bit further north, and Lemon Bay, down through Charlotte Harbor, Pine Island Sound, the Caloosahatchee River Estuary, San Carlos Bay, and Estero Bay. So historically, seagrasses within this region did not experience some of the declines that were seen in other more developed estuaries back in the 1990s. However, as many of you have as well, based on recent seagrass losses experienced throughout this region, these estuaries appear to be a little bit less resilient when impacted than they had in the past. So again, while all of these estuaries and watersheds within the region are uniquely situated with different hydrologic inputs and may have been impacted in varied ways, I'm going to share today some shared issues that have been found throughout. So I'm going to talk about what information we do have with regards to seagrass so far based on data available to the partnership, and then I will cover what we hope to be soon investigating. Okay, so there we go. Moving right along. So what are the ways our region is evaluating estuary health? Similar to other programs, members of this partnership examine water quality and clarity, sampling trend analysis, seagrass coverage and quality, and finally limited harmful algae bloom and macroalgae monitoring. Again, while each of these estuaries mentioned are uniquely situated, there are some shared issues that I'm going to go over. For one, I'm going to talk a little bit about the recent declines between 2018 and 2020 and how that's reflected in the data that we have for these regions. We have both qualitative observations for multiple agencies as well as reports from anglers in our region seeing overall thinner seagrass, thicker algae, and in some cases murkier water. And again, these declines followed some episodic events in the region, which everybody is aware of, 2017 Hurricane Irma, extended red tide blooms, and one thing that is also unique to our region is the cyanobacteria blooms that were seen on the Caloosahatchee River. And finally, in 2019-2020, macroalgae blooms to follow. So Again, these seagrass meadows seem to have been severely impacted following a lot of these events. These areas have received multiple stressors, but have been slow to recover. And with increased frequency of these types of events and nutrients inputs, it's pretty clear that the system is under significant stress. So I'm going to give a regional overview of the data and trends, and then share some estuary-specific information that reinforces the overall regional narrative and hopefully next steps. Okay, so here are a few of the questions that we hope to answer with the current and planned monitoring and analysis. In addition to nutrient levels and chlorophyll concentration and water quality, seagrass and macroalgae growth are also objective indicators of bay health. Obviously, they have complex interactive cycles, which are affected by rainfall, temperature, salinity, as well as other factors. I'm not going to get into all of those today. That's some of the future work that we hope to do. We are going to talk a little bit about nutrient levels, which can stimulate the growth of marine algae and result in reduced water clarity throughout. Our main focus on seagrass management up to this point has been on water clarity and light availability, but overall we believe that all the existing data in the region needs to start to be pulled together and analyzed in a more holistic way. We have begun this process pulling data into one spot on the CHNEP Water Atlas, where we can look at spatial coverage of the seagrass, as well as water quality data and even some algae data all together in one spot. So in the following slides, I'm going to go over water quality and seagrass data that have been recently added to the Water Atlas. And then we're going to continue to move. It's not all there. We're right now just looking at overall trends in basically species and what's going on with percent cover and deep edge, but we would like to continue moving in this direction by adding in epiphyte data, algae data, as well as some of that's already been collected. Okay, so starting by sharing some recent reports on regional nutrient pollution. Calusa Waterkeeper published an assessment of Southwest Florida waters based on FDEP records and databases. The report concluded that for the most part, Southwest Florida waterways are not getting any cleaner. The purpose of the assessment was to summarize impairments throughout Southwest Florida estuaries, using those 6 metrics that you can see there on that graphic. And then ranking counties within the region from increased rate of impairment from highest to lowest. And you can see that there are some trends here where there's some rapid impairment in areas that there hadn't been previously. And so again, we want to continue to look at this as a contributing factor when we're looking at overall water quality management and estuarine health. And again, this is sort of a graphic depicting, again, what is going on with impairments in our region, specifically looking at total nitrogen. Okay, and then moving right along, this graphic on the left shows map changes in seagrass acreage and location from 2018 to 2020 throughout our region. So I just wanted to mention this is actually just in the northern part of our region. So when you start to look at Pine Island Sound, Matlacha Pass, and south, Caloosahatchee Estuary and Estero Bay, as well. So shared some of the data that's going on with the Water Management District. They are doing some current mapping. The maps that we have now for that region are from 2014, so we don't have the more recent changes. So when those soon-to-be released maps are updated, we will be adding that to our collection of maps on the Seagrass Water Atlas, and we'll be adding that to our analysis for helping our overall understanding of progress in the region. So today, when we're looking at these southern estuaries, we're only going to be looking at the Transect data that was captured in recent years. On the right, you can see that actually CHNP estuaries throughout have a really great comprehensive monitoring program from the Aquatic Preserves from FDEP. And so you can see there's around 5 to 6 sites, monitoring sites throughout each base segment, looking at the different, and again, these base segments are divided based on hydrological inputs. And so we're going to be kind of examining some of that data. The Aquatic Preserves made that available, and we're going to be pulling that onto the Water Atlas, and I'll be sharing with you all what that looks like. Okay, and then finally, looking at macroalgae. There's no one entity in any of the CHNEP estuaries appointed to collect macroalgae data on its own, which is, one of the things that we discovered during our recent macroalgae summit. However, there are some great existing sources for historical data to give us some context, and there is some emerging new data collection as well. So I have examples of each of those here. So on the left, you can see the Fisheries Independent Monitoring data from FWRI. And again, their data is focused on fisheries monitoring, but they also collect a number of other parameters when they are collecting the fisheries data, one of those parameters being algae bycatch. And so you can see here, giving historical context, each of those arrows represents when each of those gear types they began use of those gear types in the Charlotte Harbor estuaries. And so we have data stretching all the way back to the early 90s, and you can see we didn't start to see massive amounts of macroalgae in our region until almost 2012, according to that historic data, which is great at kind of giving us a little bit of context of what's going on. But moving forward, if we really want to better understand macroalgae, we need to establish more frequent monitoring programs. Luckily, we do again have the essentially bycatch data that's also collected by the Aquatic Preserves at the Transect sites. And we also have the Sea Grant Seagrass Citizen Science Seagrass Monitoring Program that recently kicked off and is moving into, Betty had just mentioned, is moving into more of a long-term monitoring. So that's going to be ongoing data that we can add to sort of our collection of information that we have when trying to understand the region. So this is actually data that was collected by Sea Grant, FDEP, and a couple other agencies, I believe the Water Management District, when they went out to look at macroalgae percent cover when we were having this massive bloom in the Charlotte Harbor region. So you can see there, we do have a couple different sources for it existing, but this is where we're making our expansions based on recent decisions by members of the partnership. And finally, we have other considerations as well when we're examining estuary health. We need to keep this in mind, that this particular region of Florida is influenced by episodic ongoing red tide events, also cyanobacteria blue-green algae blooms that travel south from Lake Okeechobee out through the Caloosahatchee River. And finally, we need to keep track of circulation and storm events and what's going on there. So essentially the next few slides, I'm going to move down through each of the estuaries and show you similar data for each of our Charlotte Harbor estuaries, and then kind of circle back and talk about some overall trends. So I'm starting out with coastal Venice estuaries, which is Donna and Roberts Bay. And you can see here, this is what we have so far on what's going on with nutrient pollution in the region. And so there I have the FDEP, impaired waters rule assessment, like what that actual region's impaired for. And then we also shared the water quality report card from Conservancy of Southwest Florida. And we had previously also shared with you the Calusa Waterkeeper report card on this region. Overall, what are we seeing? Seagrass is decreasing, algae is increasing, and we do have some nutrient issues in this area. And that map there on the right just shows 2018 seagrass in the green, and then or 2020 seagrass in the green, and then basically the losses experienced between 2018 and 2020 in those estuaries. So the Donna and Roberts Bay seagrass areas, this is a pretty highly colored system, so a lot of the seagrass in this region is focused right around the Venice Inlet, and so you can see it's not a high amount of acreage that's already existing here, but there is a lot of fluctuation of acreage. So on this graphic, we're depicting basically what's on the water atlas now, which is the seagrass acreage variation since 1988 in the region, and then some of the monitoring data we have. Now this data looks a little bit different than the data that I'm going to share throughout the rest of our regions because we do not have aquatic preserve data for this region, but luckily we have this great Sarasota County data for this region. So the one difference that I will note is that there is not deep edge data for this region, but we can look instead at total percent seagrass coverage and then percent coverage by species. And you can see that looking at this together, when you're looking at what's going on with the species coverage and the total percent cover, and then you're looking at that together with the acreage data, you can really start to see the changes that are reflected in the acreage data a little bit early in some of this monitoring data. So it's great that we have these data sources. And again, pulling this all together is a key step that we hadn't had the capability of making until now, you know, just funding and resources and whatnot. So, you know, again, all of these agencies have different parameters that they're looking at and different priorities for how to be looking at those things. So that's what we're trying to do as a partnership going forward, is to start, let's pull all the data together and look at it and start to evaluate overall trends. And again, algae data for this region, thank you to Sarasota County. We have some great algae data, and you can see there, they collect it throughout all of the bays that are in Sarasota County. Again, we're focused here over to the right on Don and Roberts Bays. You see basically decreasing trends in seagrass and increasing trends in epiphytes and algae. Moving down through Lemon Bay estuaries, again, you see some increases in nutrients. You see nutrient impairments throughout this region. It doesn't have a great water quality report card grade. That's based on a number of different factors. I invite you all to examine that report as well from Conservancy of Southwest Florida. It looks at, again, pollutant loading. It looks at pervious surfaces. It looks at development and a lot of other things that are go into the evaluation of that report card grade. And again, so the map here on the right is Lemon Bay seagrass loss between 2018 and 2020. And so you can kind of see it's throughout the region, but again, that's why we like to look at this data with the monitoring data. So now we're getting into some of the aquatic preserves data that we have long term. And you can see here that acreage didn't just start declining in 2018, it actually started in 2016. And what we understood with the aerial data, aerial extent data, is that the losses that we were seeing between 2016 and 2018 were focused on the northern portions of Lemon Bay, upper Lemon Bay, specifically around where the Venice Canal flushes out into Lemon Bay, upper Lemon Bay, and Alligator Creek. And that area doesn't receive as much tidal flushing, and so was a little less resilient than some of the lower Lemon Bay areas. So it seemed like those seagrass losses got to those areas earlier, and then seagrass loss sort of happened throughout the entire region in that 2018 to 2020 region. So it wasn't just following the storm event, there was already something kind of going on in this region. And so again, we're looking at the seagrass data as related to Thalassia and Ceragonium. We picked a couple of different species in this region, and you can see that there are decreases in all across the board that are reflected in the Transect data, this quality data, as well as in the acreage data, and that's why we like to look at this together. But there's two things that I want to note that we're going to start to see in this Transect data is that we are seeing sort of a, I don't want to say like a full bounce back, but some recovery in this 2020 data that's already available. And that's why it's great to have this annual monitoring, is that we did see some pretty crazy declines in this region, but we're already starting to see how seagrasses are bouncing back. The other thing of note is looking at this deep edge data. It doesn't seem to match that the deep edge is actually seagrass is growing more shallow as we're losing seagrasses, which is indicating that this may not just be a function of water clarity at the deep edge throughout the water column and light availability, but perhaps this was a limitation of light throughout the seagrass beds, perhaps related to epiphyte coverage and shading from macroalgae, as was mentioned in an earlier presentation as well. And you're going to see that pretty much throughout all of the region. You don't see this steep decline in deep edge along with the decrease in seagrass. In fact, some areas you see the deep edge actually increases the depth at which it grows in opposition to what's going on with the seagrass in the region. So that's why, again, we think that we need to be starting to look at this data a little bit more holistically. And again, looking at the algae data and its correlations. And again, you see this steep increase in macroalgae data along with a decrease in seagrass in this region. Okay, so moving down through Charlotte Harbor Estuary. Charlotte Harbor is the largest, deepest, and most diverse of the estuaries in this region. It's influenced by Peace and Myakka rivers, as well as the Charlotte Harbor proper watershed. And you'll see also there's seagrass loss throughout when you look at that map on the right. It's not just focused on the upper harbor areas that are influenced much more highly by the rivers, but it's also in this Gasparilla Sound region, which is over here on the left, which is actually way more influenced by the presence of Boca Grande Pass and Little Gasparilla Pass that are right there. But you see seagrass loss throughout these areas as well. So this is indicating that maybe it's not just, and there might be different reasons for these, obviously. This isn't, I'm not trying to make the overall definitive statement on what's happening with seagrass loss in our region, because there are, again, different hydrologic inputs in all of these different areas, but we are just seeing these common themes throughout. And there is seagrass loss throughout the region. And again, increased algae in all of these areas. And we also shared some of the increased verified impairments in this area from the Calusa Waterkeeper report in that little graphic there in the center. And so looking at the Transect data along with the acreage data, you can see here the dramatic acreage loss and that graphic on the upper left. And then we pulled the Transect data from the lower east part of Charlotte Harbor. Again, this is that east wall over there on the right of the graphic, just to give you all some context. So you can see when you're looking at the west wall, you can see that there is some deep edge loss. But when you look at the wall in the east, the seagrass loss is actually a little bit further towards the shoreline. And so that's why we pulled this data into this slide, so you could sort of look at that along with what's going on with the acreage data. And that was the area that experienced the most devastating seagrass loss. Those were not ephemeral haliduli beds. Those were well-established thalassia beds that were in that region. And you can see pretty severe declines of that thalassia when you're looking down at the graphic there, 2018 and 2019. However, there is some indication of recovery in that 2020 data, and that was also shared by Aquatic Preserves staff. So we're really happy to have this data sort of up in a shareable manner on our water atlas for everybody to view. And again, we also shared what's going on with the deep edge in the region. So speaking about new data, this is some of the macroalgae and seagrass abundance data that was collected by the Citizen Science Monitoring Eyes on Seagrass project that was kicked off in 2019. And you can see here again, so the east wall is that E1 and E2, that was that Transect data that I shared earlier. And so you can see here spring and summer of 2019, you have a lot of macroalgae in those regions. And when you look at the bottom portion of these graphs, again, there is some macroalgae in spring of 2021. And you can start to see the shift back to seagrass in the summer of 2021 that you didn't see. There was sort of a different balance looking at summer of 2019, spring of 2019, and you start to see that balance start to shift here in 2021. Again, another indicator that this system is slowly recovering from some of those impactful episodic events. All right, so moving down through the southern portions of the CH&EP estuaries, again, we don't have the overall acreage data that we're going to be looking at here, and so instead we're going to focus mostly on what's going on with the Transect data. And again, it's great that we have this really comprehensive data throughout the region. So Pine Island Sound, Matlacha Pass, and San Carlos Bay estuaries are here all grouped together. Again, they sometimes act a little bit differently. As you can see, that Pine Island Sound has a lot more tidal flushing. There's 3 passes right there. Matlacha receives some more inputs from the Caloosahatchee River estuary as well. And so we actually pull apart that Transect data so that we can really tell what's going on with those different seagrass beds in the region. And again, just sharing some of what's going on with water quality in the area. We see increased impacts from nutrients, decreased seagrass, and increased algae. So the top 3 graphics depict what's going on with seagrass in Pine Island Sound, and the bottom 3 are what's going on with seagrass in Matlacha Pass. So you can see here that the Matlacha Pass region is a little bit less perhaps resilient than Pine Island Sound. There's a little bit less tidal flushing, and so it's going to be much more impacted by freshwater inputs, large inputs from nutrients, and maybe not be able to bounce back quite as quickly. And that seems to be reflected here in the data, but they do correlate, as you can see. And again, we see those increases in 2020. So a little bit of throughout the region. And so macroalgae monitoring data, there is some data collected again by the Aquatic Preserves, and I'm going to share a little bit more about some new studies that are being conducted by SCCF in the region, FGCU, to better quantify what's going on with macroalgae in this region, specifically Matlacha Pass, which has seen multiple different types of these monoculture blooms. So again, looking at the Caloosahatchee River Estuary, these are the 3 counties that surround the entire Caloosahatchee Estuary, going all the way up through the freshwater portions as well. And again, we see increased impairments, nutrients, and again, bacteria impairments, and it also has a low water quality grade overall. And Caloosahatchee seagrass transect data. Again, they also capture deep edge and what's going on with species and what's going on overall with total mean abundance. So species abundance and then total mean abundance. So we actually, the trends here are a little bit different than what we've seen throughout the other regions, but again, not as much seagrass overall in this region. And what we would like to do is start to, when we pull in the acreage data for the tidal Caloosahatchee when that's available, that's going to be up on the Water Atlas along with some of this to hopefully give it more context. Again, we don't know what's going on with acreage right now in this region, and so we don't have the complete picture, and when we do, there'll be a little bit more information about that available. We're also going to pull in some of the freshwater SAV data that's available for this region, as well as the Valson area that's being monitored a little bit further up the Caloosahatchee River, and that'll be some future work that we'll be sharing. So as far as algae in the Caloosahatchee, there again is cyanobacteria monitoring, but as far as macroalgae, you can see that there's a lot of macroalgae abundance at actual seagrass sites. So macroalgae appearing in areas where there's seagrass coverage, sticking to the seagrass, and then also benthic mats of macroalgae coating where there is sparse seagrass. So again, potentially shading out that seagrass that is existing in those regions. And finally, and I appreciate you all bearing with me as we move through all of our estuaries, Estero Bay Estuary, you see some of the similar nutrient trends. And here's some of that seagrass monitoring data. Again, we don't have the aerial data, the spatial data to share with you all yet, but you can see here from mean abundance and trends with species and with deep edge that, again, there did appear to be an increase in the 2018-2019 data. But when you look at Thalassia at the top right there, the 2020 seems to be bouncing back a little bit. And actually, Estero Bay Aquatic Preserves conducted some preliminary studies to understand what's going on with seagrass versus macroalgae in the region. And we'll be sharing the full report when this is shared with us when it's available. But you can also see that it seems that the macroalgae coverage is inversely related to what's going on with seagrass coverage in the region. So what can be done? We need to provide better answers regarding relative influences of stressors throughout the region and more actively address significant threats. So here we listed some of the monitoring gaps self-identified by partners working together throughout the CHNEP region. To start, CHNEP seeks to continue analysis of already existing transect data from the Aquatic Preserves in addition to what we shared today and what's already available on the Water Atlas. Additional parameters such as macroalgae and epiphyte load will be pulled onto the analysis in future work. This will help us examine not just the quantity of seagrass, but also the quality of seagrass. We want to continue to look at that all together, and to also look at that together with water quality data. We seek to investigate maybe potential source concentrations for increased nutrients to better manage our watersheds. We would like to do some hotspot analysis for nutrients and circulation within the region. We think throughout the CHNEP estuaries, there's increased evidence of nutrient pollution that we would like to continue to better understand the correlation between nutrients and this algae that we're seeing. There is increased indicators that there's not just diverse communities of healthy algae growth, but recurring mass monoculture blooms of algae when they do occur. And we'd like to better understand the requirements of these algae so we can track down the sources for these regions. Finally, we would like to explore the relationships between benthic cyanos, which we're starting to see, which had not historically been documented in our area, seagrass, macroalgae, as well as changes, in water chemistry and sediments. That being said, the 2018 to 2020 seagrass loss appears to have at least been in part a response to the series of events, Hurricane Irma, followed by red tide and macroalgae blooms. Shading from HABs and macroalgae blooms could have been a factor in this region, rather than a reaction to overall reductions of water clarity, as growth at deep edge of transects remains stable or even increased as seagrass acreage, overall abundance, acreage, and individual species declined. Seagrass also appears to be recovering in annual monitoring data. However, this does not mean we should dismiss recent events. With all those factors leading up to seagrass loss could likely happen again, with climate impacts including increased storms and nutrient runoff, as well as rising water temperatures, to name a few. And again, we would like to continue to explore some of that. We have not started to dive into temperature and salinity data as of yet. But it does seem that, following storm events in 2004 and 2005, we had Hurricane Charlie, and we did not see this huge impact to seagrasses that we saw recently. The system appeared to recover much more quickly than it did this last time. And so we continue to work with our partners to better understand causes and investigate solutions. Many thanks to our partners for the data we already do have. And I just listed here some of the examples of seagrass and water quality monitoring throughout the region. And with that, just here's some available resources on chnep.org. You can see we actually built out some seagrass fact sheets to help the public better understand, public and policymakers, what's going on in our region, as well as some of the new pages that we're rolling out on the CHNEP Water Atlas. So thank you all. I hope that, again, rolling through those estuaries, it doesn't get too repetitive, but we appreciate everyone bearing with us through there.
Thank you so much, Nicole. That's good stuff. And bridging the swift mud and the soft mud, always an interesting effort. And we appreciate the work that you and Betty and many others have done down there to keep that ball rolling. Got a good some questions in the chat. Marcus, do you want to go first?
Yeah, I just had a quick question about the macroalgae data from the Femme monitoring data set. Lauren provided an answer to that. I mean, we've had similar needs here, especially in light of what recent events have happened in Tampa Bay in 2021. I'm just curious how useful the bycatch data is from Femme for the macroalgae. One of my first question was, can you use it to differentiate seasonal patterns as opposed to just sort of long-term annual trends? And secondarily, how detailed is the taxa differentiation in that data set?
So, I mean, it's a pretty thorough data set collected throughout the year, but there is not a lot of taxa detail. So I think, and again, this is just our perspective on some of that data. It gives great historical context, and it is interesting to see when algae bycatch started occurring, but probably the aquatic preserve data would be more useful when we're looking for differentiating what's going on with species in the region.
Great, thank you.
Chris had a couple comments about west versus east walls and the longshore bars. Chris A?
Yeah, there were just more comments, but I think, you know, Nicole's slide really captured the importance of these longshore bars that, you know, they are key to, you know, maintaining the seagrass. It's no surprise that on the you know, landward side of the bar, prior to the loss, you had a lot of thalassia because, you know, those areas tend to be more stable. But I also think that Mark Walton brought up a good point in the chat that, you know, that it could be, you know, construed a bit like a double-edged sword, and that, you know, that could also explain why you tend to have this accumulation of drift algae in those areas, which, you know, in large quantities obviously can be harmful to grass. But that was just more of an observation that, you know, and also just some of the patterns that you can see along the west wall versus the east wall. I think Nicole, you know, already talked about that. You know, we saw loss on the deep edge on the west wall, as opposed to really more the loss on the east side of the harbor was more focused on the area inside of that longshore bar. But that was it. I was just kind of pointing that out.
No, I appreciate that, Chris. And it's we definitely don't want to make too sweeping of generalizations with what's going on. There are some overall overarching trends, but, you know, west and east wall do demonstrate that there's a couple different factors impacting seagrass in the region as well.
Okay. Again, thanks, Nicole. Much appreciated. We're going to move north again up to Dave Tamasco in Sarasota Bay. Let's find him and share the, give Dave the power.
Give me the power. All right.
And we're seeing your screen, Dave.
Okay?
All is good.
What's that?
All is good.
All is good. All right. So I want to thank the co-authors on this, Jay Labroni, also Chris Anastasia and Brad Furman. They've reviewed what we've got here. However, if there are any answers that I give that are not necessarily clear to them, we won't worry about it too much. So Sarasota Bay sensitive nitrogen levels, we don't need to go through the cartoon too much, but there's stuff that's captured in this cartoon we think is important, which is the management paradigm that's been used in some parts of Florida that, you know, nitrogen loads give you chlorophyll, reduces water clarity. It's more complicated in Sarasota Bay because we believe that some of the losses we've seen with seagrasses are manifested through things other than, you know, how much phytoplankton's in the water column. So we've quantified the light attenuation by epiphytic algae in Sarasota Bay 30 years ago, and it's about 50% on average. And so the minimum light requirements for seagrasses at deep edges in Sarasota Bay has always been higher than estimates from, for example, Tampa Bay. And we believe that's because there's, for some reason, more epiphytes. So we believe epiphytes are a manifestation, or can be a manifestation, of nutrient loads, just like the literature suggests. Macroalgae, some really great data sets that we've got from Sheila Scalaro, who abandoned us in Sarasota to go up to Tampa Bay, has done some really good work that shows that some of the problems we see in the lower part of the bay associated with increased nutrient loads in, say, recent years seems to manifest itself through macroalgae rather than, you know, typical phytoplankton. So when we manage Sarasota Bay, we're trying to not create the condition on the left, which might be, you know, the exumas, giant tortugas, but we don't want to be like the Indian River lagoon. We don't want to have a phytoplankton-dominated system, which is maybe where they are right now. So we're trying to get the best version of Sarasota Bay, which is kind of right in the middle. And so we don't simply rely upon numeric nutrient criteria because it's only focused on phytoplankton. And every atom of nitrogen that is in epiphytic algae or macroalgae is an atom of nitrogen that's not in the water column. You can't be in two places at one time. And so if you are stimulating macroalgae by excessive nutrient loads, you're not going to necessarily find it in the water column. So we have a report card that's going to be sensitive to all of the things that come in there. Our report card is frankly based upon data, and we're trying to get within there a reference period that we can use for our pollutant load reduction goal. We don't know what Sarasota Bay was like 100 years ago. We don't know what it was like 50 years ago. Some parts of our systems, like Tampa Bay, uses a seagrass target based upon photography from the late 1940s, early 1950s. The photography that we have in Sarasota Bay from back then doesn't allow you to see deeper than about 6 feet. Nowadays, we can see, great photography for the seagrasses, but back in the 40s and 50s, you didn't have people measuring secudus before the plane went up in the air. It was someone was shooting aerial photography for mapping out land, and what did the bay look like? Well, our photography doesn't allow us to see less than more than 6 feet of depth. So if you've heard that Sarasota Bay has more seagrass in the 1940s, just put that out of your mind. We have no idea how much seagrass we had in the 1940s because we couldn't see deeper than 6 feet. But we have good data from about 2006 to 2012 on multiple indicators: chlorophyll A, total nitrogen, seagrass, and macroalgae. Macroalgae is not everywhere, but we have good data across most of the bay. And so our reference period that we have is 2006 to 2012. And the reason why we have that is because we have actual data on all 4 of those indicators. And we also know that this was a healthier bay back then. We had a 28% increase in seagrass coverage during that time period. That doesn't happen if you're in an unhealthy bay. We had lower levels of nitrogen, where we had no bay segment impaired for chlorophyll A. We believe our chlorophyll criteria are quite good. And since we've got macroalgae data, we can show that back then, where we have the data, we had less macroalgae. So our report card includes 4 elements and actually includes a reference period that we're using for our pollutant load reduction goal. So just quickly to go through the math of how it's done. Not everyone loves a report card, but our audience is not you guys. Our audience for a lot of this report card is the general public, elected officials. But we do want people to understand where it came from. This has been through our technical advisory committee as well. So chlorophyll A and nitrogen, the higher the number, the worse it is. So what we do is we have a reference period, and then for each base segment for each year, including the reference years, we compare it to the average during that reference period. And if you have an annual geometric mean that's below the reference period mean, that's pretty good. That means you've got nitrogen or chlorophyll lower than what you had during a time period when the bay was good. That gets you a 4. If you're above it, but you're within a standard deviation, we'll give you a 3. If you're above it, but you're outside of standard deviation, we'll give you a 2. If your data are normally distributed, some of it is, not all of it is, then you should exceed the high end of plus standard deviation about once out of every 6 years or so. So if you exceed it 3 years in a row, that's really not normal. And you get a 1 if your value for your base segment for any given year is worse than the worst value during a reference period. So for seagrass, it's kind of the opposite, and we only do seagrass data every 2 years. But a high value is good. So if you're above the reference period mean, we give you a 4. If you're below it, but within a standard deviation, we give you a 3. If you're below it and outside of 1 standard deviation, you get a 2. And if you have less seagrass than the worst year for that base segment during the reference period, will get you a 1. And then macroalgae, we actually are using a data set that was collected by Sarasota County, but now we're replacing that with a data set that's based upon the work that Betty has been doing. And thank you, Betty Stogler, for helping train our intern who trained folks who we've now visited over 100 sites in Sarasota Bay to collect data on macroalgae distribution. So we include all this together. We don't weight anything more than the other. And we have 5 different base segments. And we're giving you this information so you can understand that, like, there's no single Sarasota Bay story. There's actually about 4 different nutrient management paradigms and 2 different seagrass paradigms that we have to work through. But for those of you unfamiliar with Sarasota Bay, here it is. Palmasola Bay to the north. The big part is fairly consistent water quality in upper Sarasota Bay. And then 3 very different kind of systems in the lower part of the bay. We were just out on all 3 of them yesterday even. So we spent a lot of time on water. So we color code these things because, again, our audience is, elected officials, the general public, and so the color coding is the cooler colors are good, like blue is the Gulf of Mexico, and yellow is warning, and red is like stop what you're doing. So if you look across that reference period, 2006, 2012, the bay was doing good in all parts of the bay. It wasn't driven by like one part of the bay doing well. This bay was healthier, 2006 to 2012. The blues and the greens represent the higher scores. Palmasola Bay continues to do well. The upper part of the bay was doing pretty good, but then 2017, 2018, it dips into the yellow. Things happened, but then it goes back into the green again. So the scores don't go that low, and then they bounce back up into values of 3. Remember, you get a 3 if you're within a standard deviation of the reference period. We're going to talk a little bit about that because the seagrass problems that we see in Sarasota Bay in the upper part of the bay look like the conditions that cause the problem have now been addressed, if you would, or they don't currently exist. But in the lower part of the bay, we shifted to yellow back in 2013. And we believe that part of the problem in the upper part of the bay is 2 red tides and a hurricane. 2016 red tide, 2017 hurricane, 2018 red tide. You can't blame red tides and hurricanes for what happened in Roberts Bay, Little Sarasota Bay, and Blackbird Bay, though, because it precedes that. We shift to the yellow in that time period around 2013. And even in that lower part of the bay, you can see that Roberts Bay shifts back up into the green. The scores get higher, and the lower part of the bay doesn't. So what we really have going on is we've got a different kind of scenario. We've got this kind of like upper part of the bay that shifts to yellow, comes back up. Same kind of thing in Roberts Bay, recent improvements, but yellow earlier. So we actually have kind of like 4 different things going on. Healthy system, a healthy system that was set back, a stress system with some evidence of improvement, and then ongoing stresses in the lower part of the bay. So we're going to talk about that in a little bit greater detail. So our overall indicator of how is the bay doing is seagrass. And we peaked in 2016. We're at 22% decline. Now, we're not the 60% decline over on the IRL, but we don't want to get that way. The last 2 manatees we've seen in Sarasota Bay have been fat and healthy looking and no scars even. So we're kind of happy about like the health of our manatee population. We don't want to lose more than 22% of our seagrass, but we have to understand why that happened. We have to act upon a watershed that is not natural at all. There's nothing natural about Florida's peninsular. And so there's nothing natural about our nutrient loads. We have a nutrient enriched state. And one of the things we want to do is act upon the nutrient sources that are most important to us in Sarasota Bay in particular. But even that bay-wide picture doesn't capture what's really going on. We have 2 different phenomena happening in the upper part of the bay versus the lower part of the bay. In the lower bay between 88 and 2014, we had a 30% seagrass increase, some variability in there. But then from 2014 to 2018, that's all gone. And we're back to where we were 30 years ago. The good news is we're not seeing continued decline from 2018 to 2020, but we lost all our seagrass increase from 2014 to 2018, but then it didn't continue to be lost. In the upper part of the bay, the biggest increase we had is 2006 to 2008. That's about a 2,000 acre increase we had in that system, a little bit more than that. And we didn't have that 2014 to 2018 decline, but we had a big decline between 2018 and 2020, where we don't have a decline in the lower part of the bay. So we got 2 different things going on with seagrasses in a system that's much smaller than Sarasota Bay, but we don't have one story. We have at least 2 stories with seagrass. We have about 4 stories with nutrient management. So the lower part of the bay, we believe, is more of a typical cultural eutrophication phenomenon. And we're going to talk about that for about 3 slides. And then we're going to shift to the upper part of the bay where we think we have something very different going on that requires a different type of approach. So in the lower part of the bay, the Roberts Bay water quality data is shown on the right-hand side. If we just looked at that and we ran a stat, we would find an increase, and it would be an arrow that points up. And there's nothing wrong with that. That's true. But what happened is a little bit more complicated than that. So if you remember, we shifted to that yellow category around 2013. So on the right-hand side, we got annual geometric means. We put all those 2013 onward years into that green box. That red horizontal line that we see right here, that represents the highest prior nitrogen concentration of the prior 15 years. And what we can see is 5 of the 7 years in Roberts Bay had a nitrogen concentration higher than the worst year, the prior 15. So all of those would be 1s. That's worse than the worst year. So we have a lot more nitrogen that shows up. And we also see that 4 of those 7 years we exceed our chlorophyll criteria, which suggests our chlorophyll criteria is problematic. So this kind of does indicate that, yeah, nitrogen has gone up, but it's actually more complicated than that, because the last couple of years, the nitrogen has gone down in Roberts Bay. So you can't blame this on simply like, well, we've got more people moving into the watersheds, so why did the nitrogen go down? So what we try to understand is, why do we see the patterns that we see in the bay? And the best thing for us to look at is the farthest downstream water quality station in the largest tributary. And this is Philippi Creek at Tamasco Trail. The raw data are the dots. And again, a big shout out to Sarasota County, Manatee County, and Moat Marine Lab for collecting these data. This is how you manage our system. The best way to explain mathematically what we're seeing in Philippi Creek at US 41 is the red line. It's a polynomial equation that kind of shows nitrogen going up and then going down, and that's kind of what your eyes see. So what could cause nitrogen to go up and then go down? We think it's the activities that occurred in that yellow box. That yellow box represents 5 years during which we had overflows from a wastewater treatment plant that was not an AWT. We do not have direct discharges of nitrogen from non-AWT plants in our system. Neither do you in the inner lagoon. But we had overflows that were miles away from the bay, and they were reported. They were reported every month, but the regulatory agency responsible for oversight of wastewater treatment plants didn't create the requirements to fix it. The requirement to fix this system was brought about through an administrative finding that was triggered by a local NGO. So that yellow box represents the initiation of the overflows and then the end of the overflows. To their credit, Sarasota County has stopped the overflows from this wastewater treatment plant, but it was 750 million gallons over 5 years. And it was miles away, yet nonetheless, that time period of these overflows matches up with the elevated nitrogen in the creek and the elevated nitrogen in the bay, and more macroalgae and more nitrogen is giving us more phytoplankton and less seagrass. So again, your nutrient loading model, if it doesn't capture an overflow miles away, maybe it's not capturing the thing that needs to be acted upon in this system. So that's the lower part of the bay. What we believe we need to do is to act on the continued influences that are happening in Little Sarasota Bay and Blackbird Bay, which we believe, again, they're not due to a point source. We don't have point source discharges into Sarasota Bay, but we do believe we have septic tanks, and we do believe we also have over application of reclaimed water from non-AWT plants, just like we have in lots of parts of Florida. So our nutrient increase from that reference period to this more recent time period is about 20%, or about 12 tons of DIN. We have 12 tons of DIN that we're looking to get out of the system, even though we don't have a single point source discharge. To their credit, Sarasota County has publicly committed to spending $500 million upgrading their wastewater to AWT with no direct discharges. So AWT with reclaimed water is going to be one of the best things you have in the state of Florida, and that's where we believe we'll be in the next 5 to 10 years. So that's the lower part of the bay. Upper part of the bay, something very different going on. Upper part of the bay has typically had very good water clarity, but between 2018 and 2020, we had a big seagrass decline in that area, and that's what the area in red shows. This is overall about a I think it's like a 16% decline in seagrass coverage in this time period, 18%, something like that. It's in the high teens % decline in seagrass coverage. And it's happening in a part of the bay that traditionally has good water clarity. If we look at that kind of like, scorecard of how we're doing, remember, this is multiple indicators. The lower part of the bay, as an example, in Roberts Bay, we dipped into this yellow range for years, for about 4 years, 5 years. We had multiple indicators of conditions, macroalgae, nitrogen, chlorophyll, that indicated that we were below, we were outside of a standard deviation. We were outside of what you'd expect to see in that reference period. But in the upper part of the bay, we don't really have the same kind of phenomenon, yet we had this big seagrass loss. So what we think happened in the upper part of the bay is an episodic event rather than traditional eutrophication. We're very careful with this. The language we use is that humans don't cause red tide, but we can cause them to be worse. And we believe that recent data that was done by Miles Medina supports that. And so our thing that we have to deal with in the upper part of the bay is different than a typical cultural eutrophication phenomenon. We believe, and we're going to show you the data, that what we had is a strong red tide in 2016, Hurricane Irma in 2017, and then a stronger red tide in 2018. The photography for 2018 occurred prior to the 2018 red tide impacts. The photography for 2020 occurred after the 2018 red tide impacts. So let's see if we can actually back up that paradigm with real data. This is data collected by Mote Marine Lab. The y-axis here is in 1 e to the 7. E to the 7 refers to the exponent 7. So a value of 1 actually represents 10 million, not 1 million. And what you can see here is that in 2016, the late part of 2016, the latter part of the year, the water is warmer in the fall than it is in the spring. We had some pretty high levels of Carenia brevis cell counts. But we had even higher levels in 2018, where we had values that are higher than 10 million cells per liter. So very high values, again, in the latter part of the calendar year when the water temperature is warmer. And here's our more recent 2021. So we've had 3 red tides, but the strongest one by far was the 2018 red tide. We also had in 2017, it was a hurricane. We had 100-mile-an-hour winds. But the hurricane passed to the east of Sarasota Bay, so it actually blew water out of the bay. There's pictures of manatees stranded on grass beds in Sarasota Bay. But nonetheless, we had very high turbidity in 2017, the 2018 red tide. And the red tide was actually concentrated in the areas where we had the seagrass loss. So the seagrass loss is centered right around this area. This is a very detailed Carenia brevis assessment carried out by researchers from New College. And the warmer colors are the highest cell counts. And so right here, right where we had the big seagrass loss, is our highest cell counts, right in the area of long bark line. So very high levels of Carenia. It had a devastating impact on our fish populations. This is a really neat paper if you are familiar with it or not. It's actually got links to passive acoustic listening systems. You can hear what it sounded like in the bay before the red tide and during the red tide. These represent 2 long-term hydrophones. And so you can see our fish populations declined dramatically because not only was it a red tide that killed a lot of fish, it was a red tide that made it very dark while the water was very warm. And that's very bad for seagrasses. But since then, we've had very good water clarity. This is secondary depth in feet. And you can see it's not uncommon in this part of the bay to get 12-foot visibility, 12-foot secondary depths. We have no trend of declining water clarity. We have water clarity that a lot of places would love to have. I mean, this is 8 feet. We're regularly higher than 8-foot visibility in this part of the bay. And in fact, our recent seagrass surveys, this is Jay Leveroni in the water. Here he is in about 8 feet of water, and he's about 15 feet away from the boat, and yet you can see him completely from top to bottom through the water. That's what it looks like in the bay right now. This is doing a Bras Blanquet in Sarasota Bay. And so we've got great visibility. So when we actually look at the clarity of the water in the bay, parts of it look like the Caribbean. So we've had good water clarity for a couple of years. We've not really had, this kind of long-term culture eutrophication. So how is the seagrass recovery coming from the 2018-2020 losses? And that's what we actually worked with Southwest Florida Water Management District. I want to thank Chris Anastasia and I think Mark Walton helped out a lot.
Hey,
so
Dave,
we went to 30. Yeah.
Can you wrap it up in maybe like 5 minutes or so? I hate to do that, but got 2 more presentations and we're running low on time.
Yeah, sure. Okay.
Thanks.
So what we've got is the areas of seagrass loss that we're interested in are the areas in red. And those areas in red represent the areas where we had spent a lot of time looking for seagrasses. So generally what we're seeing is we're not seeing a difference in water clarity in terms of secular depth, where we've lost seagrasses versus where we actually still had seagrass. But our Bras Blanquet scores where we lost seagrass are less than 1, representing less than 5% coverage. So what we think is happening in that particular system is we've lost a lot of seagrass and it's not coming back. We don't have a lot of macroalgae in that part of the bay, about 10% coverage. So we're not seeing these signs of culture eutrophication. We're seeing the water clarity is good. So is the seagrass going to come back? So we worked with Brad Furman. Brad Furman and his folks went out there, and what we found, let's just jump to it, is Brad sampled a bunch of sites. All the sites that we're most interested in have been processed. So how much seed bank do we see in the areas where we lost the seagrass? And the answer is hardly any at all. So we've got the water clarity back. We don't have, we don't think the typical culture eutrophication phenomena, but if the water clarity comes back, there's no seed bank, there's no below ground rhizomes. Where would the seagrass come from? So this is what it looks down there. There's some places where there's grass, but there's an awful lot of areas with good light, plenty of oxygen, lots of organisms, but we don't believe that we've got seagrass coming back. And so what we want to find out is in that upper part of the bay, in areas where the water quality appears to have recovered, does it make sense for us to do something more complicated to test out whether or not seagrass can recover? So what we're suggesting, upper part of the bay only, not necessarily appropriate anywhere else. What we want to do is an experimental transplanting at different depth gradients to find out whether or not seagrasses can now grow in this area, except for the absence of a seed bank. They would be back there anyway. And I can wrap it up with that.
Thanks for encapsulating like that, Dave. Much appreciated.
No worries.
Let's see. Chris has a question in the chat right now. Anybody else? Feel free to type them in, or once he covers his, go ahead. Chris A.
Hey, Gary, thanks. Yeah, Dave, just real quick, the graphic that you showed earlier with the seagrass over time, there's quite a jump that you see between 2000 and I think it's 5 and 08. I was just curious what your thoughts are on that, relatively rapid increase in seagrass coverage over that time period.
Yeah, we think that is basically, that's not an area where we didn't have seagrass. We think that's an area where the seagrass wasn't necessarily mapped before. So we think that what we had is we've always had seagrass there. It just became dense enough to be mapped as seagrass. And that area where we had the big gain is where we had the loss. So we don't believe that we have zero seagrass there. We just don't have enough seagrass to be mapped as seagrass. So the area of the gain that you're talking about is the epicenter of the area of loss.
Thanks, Dave.
Mm-hmm.
Any other questions for Dave right now? Okay, I'm going to turn it over to Luis Lascano Sandoval, PhD candidate at the University of South Florida. He presented at the meeting in the spring on some remote sensing, and he's got an update for us at this time.
Thanks, Gary. Okay, hello everyone. My name is Luis. I'm a PhD student at the University of South Florida. I'm gonna present real quick the seagrass mapping update from my research. So these are some of the results that I presented back in April. I basically mapped 2 regions, which were Clearwater and San Jose Sound as a single region, and Tampa Bay. So I used Landsat and Sentinel satellites for only 4 years between the 1990 and 2019 period. And I compared that with aerial data from the Southwest Florida Water Management District. We noted that there were annual variations in seagrass cover, so we really wanted to see how much of the satellite imagery available we were able to use for completing the time series. So what we have now is that we added more study regions. Now we have 4. We split Clearwater and St. Joseph Sound, and we included Sarasota, we kept Tampa Bay the same with no changes. So we explore all the satellite imagery available from Landsat 5, Landsat 7, and Landsat 8 and Sentinel-2 between 1990 and early 2021. So we can see here in this plot that the temporal coverage of each of these satellites. We found imagery with good water and atmospheric visibility for 16 years between this period of time. The continuous annual mapping was possible after 2015, given the number of images, especially from Sentinel-2, which provides a better image resolution and much more images per year than Landsat satellites. So Sentinel-2 was preferred for doing seagrass mapping after 2016. So the results, what we got here is the comparison of seagrass cover trends between the satellite and aerial products. We did not expect to get identical trends because these are two different methods, different products. As we can see in the images in the right, seagrass classifications using satellite imagery provides spatial data as raster files. That means that provides information pixel by pixel. And also the pixel size is different among the satellite products. So for example, for Landsat, the pixel size is 30 meters by 30 meters, and for Sentinel-2 is 10 meters by 10 meters. But in the case of the aerial products by the Southwest Florida Water Management District, the seagrass area information comes from vector files. It means that these are polygons. So with this being said, we knew that the differences are normal and expected. But when we look at the temporal trends, we can see that the results are similar and both data sets are correlated as we expected. We did the same for the four major Tampa Bay segments, Old Tampa Bay, Middle Tampa Bay, Lower Tampa Bay, and Hillsborough Bay, to observe the impact of water quality changes on seagrass cover at different degrees. So we expected to see larger changes in seagrass areas over time in Old Tampa Bay and Hillsborough Bay because these segments are more exposed to higher sources of contamination and river discharges in comparison to Middle Tampa Bay and Lower Tampa Bay. So this is what we can see in the plots here. After 2000, we can see that the increase in seagrass area reach higher increasing peaks in Old Tampa Bay and Hillsborough Bay. If we look at the water quality data, we find this relation between the seagrass areas and water quality changes. We can see this relation between this data. The annual averages of different water quality parameters at each Tampa Bay segment is, we consider the chlorophyll A, nitrogen, phosphorus, and turbidity. This data is collected by the Environmental Protection Commission of Hillsborough County. We can find here in this data, there are several patterns. So one is the gradient of water quality in Tampa Bay, where the Old Tampa Bay and Hillsborough Bay, which are the blue and red lines here, presents the lowest water quality conditions in comparison to Middle Tampa Bay and Lower Tampa Bay, as we initially expected. The second pattern we observe is the improvement in water quality over time. But we can see that these changes are different before and after 2000. So the water quality parameters had the highest concentration levels over before the 2000. After 2000, the decreasing trends were steadier over time. So these patterns coincided with the seagrass cover changes observed in our results. We developed this Google Earth Engine app, which anyone can use. This is for visualizing and estimating temporal and spatial seagrass cover changes in selected regions and selected years. So this is a quick demonstration of the app. Finally, with this work, we concluded that satellite seagrass mapping can produce similar results to the aerial mapping products. That Sentinel-2 imagery has big potential for doing continuous seagrass mapping. Also, we can use these methods for expanding seagrass mapping efforts in other regions of Florida, Gulf of Mexico, and the Caribbean. These are some links to the Google Earth Engine app. Another link for the presentation slides from the April meeting with more details of the methods used in this work. And also stay tuned for the publication derived from these results, which we expect to publish in the next 2-3 months in collaboration with Ed Sherwood, Gary Rollerson, Chris Anastasio, and Fran Mueller-Carrier. And that's it.
All right. Thank you very much, Luis. Any questions regarding the project?
Gary, this
Hey, Gary, I'm sorry, go ahead.
I'm going to interrupt Chris. You've been asking too many questions, so I'm going to ask some. Just a feature request, Luis. In terms of the Google Earth Engine app that you just developed, it looks great. And I was wondering, based on the imagery that's available for a particular year, is there a way to depict that in the Google Earth Engine product in some way, just to get a sense of how many images were used to derive an annual estimate over the years? That would be helpful.
Yeah, it varies a lot. It depends on what satellite product was used. So, for example, for Sentinel, we got more images per year. So we use like 5 or 6 images per year using Sentinel. But for Landsat, there were some years with just 2 or 3 images. So,
yeah, I get that, and that would be useful information to depict on that app, just so the user is aware of what how many images went in to derive a particular annual estimate.
Okay, yeah.
Just to add another comment, mostly that as we move forward and try to manage seagrasses, I think throughout Southwest Florida, you're seeing sort of how the interplay of nutrient management and then episodic events is pretty critical to our overall understanding of seagrass recovery patterns and losses. So I think tools like this, higher temporal resolution tools like this, are going to be important for us to move forward on how we manage these resources in the future. And I encourage the continued development of them, as well as looking at applications of these seagrass remotely sensed seagrass products with water quality information more at a temporally frequent scale. So looking forward to potentially developing more of these products in the future with partners in the room.
Sure, sounds great.
Yeah, I think one other thing that I noticed looking at a couple of these different products is that it seems to me like the continuous grasses are really mapped well by the remote sensing products. A lot of the non-agreement is where there's some considerable patchy seagrass beds. That's not, as far as I can tell, that's not often or not captured as consistently by the remote sensing. So that that's something to think about as we're going through this.
That's an issue with our aerial mapping as well. You know, when you start getting into those real patchy or sparse beds, it's just it's tough to resolve that signature.
Okay. Last but not least for this morning, we'll try to have some additional discussion brief afterwards, going over to Chris Anastasiou with the Swift Mud. Take it away, Chris.
Thanks, Gary. Yeah, I did have a follow-up question for Luis, but I will save it. Mine presentation shouldn't take very long. What I wanted to do today is just update you guys on where we are with our 2022 seagrass mapping. So I don't know if y'all can see my screen or not, but
we can.
Okay, awesome. So yeah, so this is going to be relatively quick, but there are some changes that we've made and some improvements that we've made that I did want to share with everyone. Just to reorient those of you who aren't familiar with our seagrass mapping program. So we map the entire coastline of the water management district from just east of Cedar Key, south to Charlotte Harbor. Every 4 years, every 2 years, we do the Suncoast region. So that's what you see here, which is Clearwater Harbor, St. Joseph Sound, south to Charlotte Harbor. So we'll be mapping this area again, starting very soon, actually. One of the things that we've changed this year is we've adjusted our flight window. For those of you familiar with our mapping protocol, typically we start the flight window on November 1st and it goes through the end of February. This year we have truncated that a bit and actually pushed the start date to the 1st of December. The reason being that over the last several mapping cycles, we've noticed that November looks a lot more like September than it does December. And so in order to try to capture that similarity in the extent of grass as we go into that, what I think is kind of the ever shortening winter dormant season, we truncated the window to December 1st. So we'll be starting a little bit later this year and likely moving forward. So that's one change that we've done. The other thing that we've done that we've changed, and I'll explain why we did this in a minute, is that we redesigned or we reconfigured our flight line. So really not that important for the end user, but I did want to just convey this, that we went through an exercise at the end of the 2020 mapping cycle and we came up with an optimized flight line schedule. What this does is a couple of things. Number one, it reduces the amount of flight time that the airplane has to be in the air, which ultimately reduces the overall cost. The other thing that it does is it allows us to take advantage of those times when we do have all the criteria met for flying. So we're not up in the air as long, not only from a cost perspective, but also just from an optimal weather condition perspective. So we have changed that. Now, the main reason why we optimize our flight lines is because this year we're moving away from the 12-inch pixel resolution and we're using a 6-inch pixel resolution imagery. And this is going to, we think, give us much higher resolution imagery. It's going to allow us to really make this. What is this? I think I just turned on the. I think I just turned on the closed captions. So sorry about that. What it's going to do though is it's going to provide us with much clearer imagery. So I think what ultimately that's going to do is it's going to give us a greater detail in this 2022 imagery. So what you see here on the screen is a comparison of the 12-inch imagery taken in 2020 in the same area with 6-inch imagery, which was actually taken from our digital orthoquad imagery that we fly every few years. And I wish I could turn off that closed captioning thing. Oh well. The other thing that we've done this year that we're changing is we've updated some of our go no-go criteria. Probably the most significant one to all of you is we have discontinued the use of Secchi disc measurements in our go no-go criteria. So we no longer are going to go out and collect Secchi disc. Many of you helped us in the past by going out and doing that almost every morning, and we don't really need to do that anymore, largely because we don't really find that the juice is really worth the squeeze in terms of the amount of work that goes into collecting those measurements and then ultimately using those in our decision-making process. So we're not doing Secchi anymore. We've also removed a requirement for rainfall. So we had a requirement that said that you had to have less than 1 inch of rain in the preceding 72 hours before you could give the thumbs up for flight. We've discontinued that as well because what we found is that rainfall really doesn't have that big of an impact at this time of the year. If you have a cold front and even if it dumps an inch of rain, what we found during the last mapping cycle is that it didn't really affect the clarity enough to ground the flight crew and the aircraft. So this time we're not going to use that as a go no go criteria. That being said, if we do see a large rainfall event, obviously that's going to weigh into our decision. But really, what you see on the screen right now is what is going to be driving our go no go criteria. So sun angle and tide condition, that's always been part of our criteria. Obviously, cloud cover is an important one, and wave height and wind conditions to continue to be major criteria for giving us the go ahead for flight. Another thing that we've started to do in 2020, and we're really embracing this in 2022, is using underwater video. So as you all know, because a lot of you have helped us in this, field verification is really important in order to produce a good quality map. So we're always looking for ways to optimize our field work in order to get the best bang for our buck. And so one of the things we're starting to do is really rely heavily on underwater video. We did this largely in 2020, and now we're really going forward in 2022 to collect underwater video at as many points as we can. And this has a couple of advantages. Number one, it allows us to not spend so much time on station when we're collecting data. Number two, what it does is it allows us to take the video back to the office, and we can get more eyes on the video, and we can take our time and better characterize what we see in the field. So it gives us better quality data, and it also allows us to go out and do more data. What you see in this picture is my colleague Will with his fish finder on his head. This is one of our new ROVs that we're starting to use, and it's a very cool tool because it actually allows you to guide the vehicle, which you see on the bottom center of the screen with your head, which is really cool. Anyway, one of our new tools that I think is going to make our maps even better. The other thing too is special areas of concern. So this slide shouldn't be new to most of you who are familiar with our mapping program. Passes are always a problem because of the turbidity issues. So we have these special areas of concern that will continue to be areas of concern for us. The other is these larger issues that we've talked about for most of the morning, which are these areas that we know we've had big changes. Old Tampa Bay, we talked about. Upper Sarasota Bay, Dave talked about. Nicole talked about Lemon Bay and the east wall of Charlotte Harbor. There are several areas like this. And so really what I wanted to do was put a call out to all of you to provide us with any information that you all have in these areas. Any area that you have of concern, let us know, because this is the time that we want to go out and start collecting these data. If you guys are out collecting data, by all means, you know, we would love to share that data. Marcus, I know, is on, so I always take this time to plug open science as not only as a concept, but as a culture. So if you guys are collecting any kind of seagrass data, please feel free to share that with us. And if you have any special areas of concern that we haven't already talked about, and I've worked with many of you on this, so please make sure to let us know so we can go out there and check those areas out while, or at least during or slightly after we're out collecting the imagery. If you are out collecting field data, I'm not telling you how to do it, but I'm just sharing with you all some of the things that really are important to us. So if you are collecting some data, obviously location is really important. If you see something like algal, macroalgae in areas where you typically don't see it, or you're seeing changes happening, Location's really important. So the more accurate the location, the better for us. Percent cover is actually really important too, even if it's just an aerial estimate. It doesn't have to be something as fancy as a quadrat or anything like that, or a Brumble and K-score. If it's just an estimate of percent cover of whether it's seagrass, attached algae, drift algae, whatever it may be, that's really good stuff, and we would love to get that information from you. Epiphyte loads, species, all that good stuff. And of course, you know, a photo is worth 1,000 words. A video is probably worth 10,000 words. So if you have that, please feel free to share that as well. That's really it. That's the end of my update. So there's my contact information for those of you who don't have it. Please reach out if you have any questions, and we'll be happy to get you the answers that you need. Thanks. That's it.
Thank you, Chris. Any questions for Chris at this moment? I'll just make a comment real quick that our Seagrass Transect data most is, I think 99% has been collected. We've actually been having problems getting out to ours because McDill has a firing range that's out there that they keep turning on, and we haven't been able to access it, but all of that should be available fairly soon. And so Chris will be able to, you know, do some comparisons from over the last few years to look at significant changes, especially between 2020 and 2021.
Yeah, and I'm glad you brought that up, Gary, because I would be remiss as to, you know, I always also plug the importance of Transect data, field data that Sarasota County does. I mean, that's really critical too, in order for us to provide you all with the best possible map. So I appreciate that.
Yep. Mindy Brown has a question. Do you want to go ahead and express it, Mindy?
Perfect, Chris. Yeah, I heard you say you don't need SECI measurements anymore, but will you still need people to go out on site every morning to get the site-specific, like weather, water conditions?
No, we're not. We're moving away from having people go out every day and collect any measurements. Part of the reason for that is just, you know, you're collecting data at one point. So we found that to be of limited value for the 2020 map. So we're relying very heavily on the previous weather conditions. So, you know, winds play a major role. You know, having said that, Mindy, though, you know, if there is something unusual going on, or if, you know, you're out doing your work and you find that, man, you know, the water clarity here is really bad. Yeah, absolutely. That kind of information we need, but we're not going out on a daily basis like we did last time.
Okay, that makes sense. Thank you. Yeah, we'll let you know if you see anything abnormal.
Awesome. Thanks.
So, Chris, quick question. With that more resolute imagery, do you suspect, particularly this year with some of the anomalies we had, if you're running into a lot of areas with drift algae, are you going to be more heavily relying on some of the ground truthing if you can't see the seagrasses with that enhanced imagery?
Yeah, I mean, the drift algae is always problematic, especially in areas where you have grasses that are underneath, because we only see, you know, what's at the surface. So we have those areas identified from the 2020 mapping efforts where we know that drift algae has been an issue. And so we will likely hit those areas fairly hard in terms of, you know, field verification to make sure that, you know, what we're seeing is reflective of what's out there. But ultimately, you know, we have to make the call. Whatever the signature is what, you know, we have to make that judgment call. But yeah, certainly we're going to weight some of the field work towards those areas that are known to be problematic with respect to drift algae. You know, there's one thing too I just wanted to add, and I didn't put it in the slide, but our time frame for turning these maps around has been compressed. Ed, I know you have been.
I didn't ask you yet.
I know, and I know you were going to, so that's why I figured I'd go ahead and just preempt your question. Our timeline right now, we've done a lot to try to reduce the turnaround time, and I think, you know, we've made some improvements in terms of our processes, that what we're shooting for is to begin the draft map production no later than mid 2022. So the idea is to have draft maps, at least for some of the estuaries, sometime later in 2022, rather than waiting till 23 to get the release. So expect that to be a much shorter time frame in terms of us getting you guys some numbers, even if it's preliminary. We're really trying hard to get that out as soon as possible, so that so we don't have to wait that long this time.
We definitely appreciate that, and the district being responsive to some of those comments in the past. To add another area as a potential lookout for this year, north of Emerson Point and basically on the entrance into Terracea Bay, we've seen pretty significant ulva macroalgae. At least probably last time I was out there, but might cause some issues in terms of mapping into this winter to be on the lookout for.
Thanks, Ed. Yeah, I'm writing that down now.
So we've only got a couple minutes left till the top of the hour, the 3 hours we allocated for this. I really appreciate all the presenters and the time and effort they put into their presentations. I'd like to take a couple of moments, if we could, to think about some of the ways, it actually sounds like we're reasonably well aligned with a lot of the work that we're doing, and I think we knew that because we've been at this for quite some time now. But are there what if you can put in the chat or just pipe up some of the different ways that you think we could continue to improve the monitoring to make it more comfortable, if that's a need that you think would be useful for us. And I think this kind of builds off the macroalgae workshop that Darcy Young and Sarasota Bay led this spring. And also I'd like to think about the importance of episodic event sampling. Is that something we need to potentially ramp up? Is look at what's happening after some of these big hurricane events or other stochastic events that may have a significant impact on our systems. And I'm going to really quick just put a plug in for an old Tampa Bay actionable science meeting. Some of you hopefully are on our constant contact list and received information about this. It's this afternoon at 1:30. We're talking about specifically the issues that are going on in old Tampa Bay that are directly impacting our seagrasses and how we can potentially mitigate pyridinium. But like I said, I'd like to have some quick discussions about ways we may need, do we need to align our monitoring efforts more than they are now, and how do we go about doing that? And I know that's a lot to unpack in like 5 minutes.
I'll chime in real quick. I think John Ryan asked the question, I responded to it in the chat, is, especially in response to a lot of things we experienced this year, is, a lot of times we're monitoring the symptoms of sort of loading events, and we will then piece together sort of the loadings after the fact. And I think, as a community, we would do ourselves a much better favor if we actually had an understanding of real-time nutrient loadings into the system, whether they're from stormwater point source sources or the collective sources that contribute to the nutrient pool within our estuaries. And that in my mind is a big gap that we have in comparison to other areas of the country. Again, we're sort of piecing this information together, I think, a lot of times after the fact, and monitoring the symptoms and then trying to make these correlations. So I would as a region, as one voice, try to elicit to policymakers and decision makers the importance of understanding nutrient loads from all the sources available to our coastal environment. And if we could do a better job of monitoring those through time, I think we'll be much quicker to respond to events or episodic events where we're seeing impacts to the ecosystem. So I'll put that out there sort of as food for thought and fodder.
Ed, along those lines, one of the things we're doing with our loading model is we're looking at both total nitrogen and also dissolved inorganic nitrogen. And some of these sources become Much more important if you look at DIN loads. We're a fairly well flushed system, and in much of Sarasota Bay, the amount of time that a water mass would sit in the bay is not enough for nitrogen to be liberated from a complex organic molecule. So we think the DIN load might be more important. Not that TN loads in general aren't, but things like reclaimed water, septic tanks, and spills become more important as a DIN load percentage than they are for a TN percentage. So not suggesting everyone does it, but that's what we're doing.
So I think one of the things that I've always, and I think there was some chat about the contribution of that internal cycling. I think Chuck talked a lot about that, but when we have these episodic events and you have like a lot of fish kills, there's a lot of focus on the beaches and capturing and getting rid of the dead fish on the beach. But what would be really interesting is to get a better handle on what is that potential nutrient load in the water in areas away from population centers or away from beaches, so that we can more accurately represent the potential fertilization, if you will, of a water body as a result of a massive fish kill.
And if you do the fish kill, you gotta make the conversion to dry weight, is what we found. We went through this in Sarasota Bay with the red tide. You can't use a wet weight estimate and a nitrogen content if it's based on dry weight. And the difference is a factor of 7 or 5 or something like that. So you have to make everything in the same units. Otherwise it's not comparable.
Will Van Gelder's got a question in the chat. You want to talk about that, Will?
Yeah, sure. Just, dealing with the seagrass as much as we do, and we get into a lot of conversations of seagrass, one thing that I find that may be a benefit is if we look at it from the perspective of algae and not seagrass, and just knowing the dynamics of, I wouldn't say algae is our enemy, but certainly something that greatly impacts the health of seagrass meadows and all of our estuaries. And maybe there's some dynamics that are inherent to the study of the microbial community and specifically algae that may shed light that we, don't normally look at. So just a way of looking at the balance from a different perspective.
Thank you for that.
Yeah, I think along the same lines, when we talk about sediments, chemistry and sediment dynamics, and that coupling between the benthic, structure and the water column is really important. So I think we have a tendency to focus on the water column a little too much and what's going on there, and we tend to miss the larger picture. I mean, certainly with Old Tampa Bay and that shift from seagrass to caulerpa, there's a lot happening, in the benthic part of the environment, where if you're just looking at water quality indicators in the water column, you might miss that.
Yeah, I think along those lines, I think we need to be really careful about where our stuff leads to. So for example, people have been talking about sediments as a nutrient source for lakes for decades. And they've been acting on it, but it's really hard to find a success story of a lake that got better because you removed the sediments. And I just think that, as we move forward there, realize every source that you talk about is going to lead potentially to an action. So I'm not saying it's a bad idea to get rid of sediments, but we did not get rid of the mucky sediments in the bottom of Hillsborough Bay. They burned themselves out once the points were reduced. And so just be aware of, you can spend a lot of money, get a lot of muck out of a lot of locations. Is there a success story that you can point to that said, that worked, that's why I'm doing it here as well? We have that for point sources, absolutely. We have it for the more concentrated forms of non-point sources from ag and things like that. But as you talk about sediments, just keep in mind, further down the road, someone's going to say, well, therefore I need to dredge my canal, I need to dredge my bay. Well, okay, where's the success story that you can say that did have a benefit? Look to the lake literature and you'll find that there's not a lot of success stories of positive system responses to sediment removal.
That was a question, I know Chuck had to go, but that was a question I was going to ask Chuck, because one of his slides where he showed the muck accumulation in the upper part of the IRL, I'm like, so that, does that mean we need to remove it?
Well, if you have 8 feet of muck, right, is the bottom 7.5 feet contributing to it? Maybe not. If you take 8 feet of muck, leave 6 inches, is that enough of an influence as the 8 feet? Or did you just make it deeper so it's more anoxic? So it's just something to be aware of. But think about the implications that you're finding. You could make it worse by removing 90% of the muck in deeper areas by making it deeper and not really effectively changing the nutrient supply. Because that nitrogen molecule that's like 7 feet buried underneath the muck is not going to come into the water column. It's just going to be that top couple of inches. So if you just make that top couple of inches 7 feet deeper, maybe you didn't make it better. And that's kind of like where the lake literature is.
Well, again, thank you to all of our presenters, greatly appreciated. Over the next couple of weeks, I'll try to encapsulate a lot of the conversation that we had and send that back, send that out to everyone. And I'll put a plug in for our meeting this afternoon on Old Tampa Bay Actionable Science, and a reminder for many of you out there that we do have a rescheduled basis February 28th to March 4, and we can definitely continue that conversation during breaks and at bars on into the night there as well. So thank you all, and if I don't see you before then, have a great holiday.
Thanks a lot, Gary.
Thanks, Gary. Thanks, everybody.